Day. We're very pleased that so many of you could join us for our conference today. I'm Mark McClellan, and I'd like to welcome you to our second annual State of Biomedical Innovation Conference. This is part of an ongoing series of innovation convened through the Brookings Initiative on Innovation and Value in Healthcare with generous support from the Irene Diamond Fund, finding new and better ways to bring better treatments to patients with a particular focus not just on discovery, but on developing and using valuable new treatments effectively was a real priority for Irene Diamond, and that priority is reflected in the focus of this innovation series here at Brookings. Today's discussion and the materials that our speakers will present are going to help us set the stage for ensuring that policies aimed at improving innovation are focusing on where they can make the biggest difference. Now, a few notes before we get started today. Our event is public. We do have press present. The event is also being webcast live and recorded, so everything today will be on the record. You all should have gotten a copy of today's slides when you came in the room. The monitors up at the front are a little bit hard to see, especially from the back. If you don't have a copy of those, you can get them back out at the front. And the slides will be available as a PDF on the upper right side of the screen if you're joining us for the webcast. If you'd like to revisit any part of today's discussion, the event will be archived in its entirety on the Brookings website. We'd just like to remind our panelists to be mindful of John Berard, who's up in the front with our timekeeping. And throughout the morning, we're going to have some opportunities for all of you in the audience to ask questions. If you have a question or comment at those times in each of the sessions, we'll have a microphone brought to you to speak. So we are talking about biomedical innovation today, and as I mentioned previously, this is our second annual State of Biomedical Innovation Conference. It's part of our ongoing work on biomedical innovation. Many studies have shown the impact of investments in biomedical research and development on health through new treatments and new knowledge about how to use them effectively have been substantial. But as our presenters are going to show once again, the biomedical innovation process can be costly, it can be time-consuming, and many of these investments do not translate quickly, if at all, into better health for patients. And so that's the main set of topics that we want to cover today. We're going to highlight recent trends in the development of innovative treatments and interventions. We're going to explore some novel policy solutions as well as private sector initiatives, collaborative and individual, to the challenges facing medical product development. And we aim to expand the policy discussion around innovation through looking at metrics of how we're doing. Now, this is something I'm going to introduce over the next few minutes, kind of the current state of the art of measuring progress and productivity in biomedical innovation. But as you'll hear in the discussion later today, there are certainly examples for doing more and gaining more insights here as well. So that's a little bit of an overview of what we're going to cover today. And so I'm going to start with some of the most familiar metrics of progress in applied biomedical innovation. One of these is the annual statistics or ongoing statistics of trends in new drug approvals in the United States. Almost any discussion of biomedical innovation looks at what's technically the FDA new molecular entity and therapeutic biologic approvals. On this count, there has been a significant increase over the last several years. 2012 in particular was an unusually high year, reflecting a trend over the last couple of years with 39 new products entering the market. While it's still early in 2013, there's been some questions about what's happening right now in the second quarter. It's likely that a substantial number of new products are going to be approved in 2013 as well. So this is one of the most traditional measures of industry productivity, but it also has some limitations in understanding what's going on and in terms of looking forward and the impact of the new treatments themselves as well. Many groups also track closely the financial inputs necessary for a strong product development pipeline. This chart shows, or this set of charts shows, three different funding streams that reflect contributions by the NIH, particularly in preclinical and translational and some clinical studies, contributions from private pharmaceutical industry, pharma, R&D expenditures in the middle slide, and venture capital funding for early stage and later stage clinical investments on the right-hand side. NIH appropriation levels, while doubling between 1997 and 2002, have leveled off in more recent years. We've seen a plateau in NIH funding levels with 2012 enacted level at around $31 billion. The pharmaceutical manufacturer investment, which comes from pharma, they track their company's research and development and expenditures through a detailed survey system, showed 2012 expenditures at $48.5 billion. That's reflected, again, an increase in past years, but more of a leveling off in recent years. Groups like Evaluate Biopharma have tracked the venture capital investment in biomedical space, and their analysis shows a marked decrease in funding over the past 5 years, from around $6 billion in 2007 down to $4 billion in 2012, and we'll talk more about this later on today as well. The increasing cost of research and development, something that some have associated with some of the slowdowns in the trends in investment, are reflected in this slide. This is sort of a foundation for a measure of industry productivity. If you think about the investments going in and the cost of producing each new medication on average, that gets to a measure of productivity. Many academic studies of average development cost per approved new drug product have been conducted over the last decade. These are methodologically difficult studies in the sense that how you count things like discount rates given the long time frame of investments required and how you attribute costs that are made in terms of overall investment to particular drugs coming out at particular times can have an impact on the numbers. But there are some consistent trends in these results. One of the most current studies undertaken by the Office of Health Economics in the United Kingdom pegged the average cost of development per product at $1.5 billion in 2011 dollars. This is a little bit different than some previous studies, such as by Professor Damasi and others in 2003, which came to a figure of $1 billion, and a more recent study by Dr. Paul and others, which gave a figure of $1.9 billion per product. As the chart shows, to the extent that while all these studies are not using the same methods, they do seem to be showing a consistent pattern of increases in costs over time. There have also been studies of the time it takes to develop new products, and this average time as well seems to be substantial and perhaps increasing. For example, that same 2012 Office of Health Economics report that I talked about on the previous slide found an average of 11.5 years of development time per product. A study by Ken Caton, who we'll hear from later, and Professor Damasi in 2011 added a further layer to this kind of timeline analysis by breaking down these development times by therapeutic area. And as you can see from this chart, products for neurologic conditions, CNS conditions, approved between 2005 and 2009 had the longest mean development time in this period, about twice as long as for AIDS, HIV antiviral treatments, which averaged only around 5 years. Putting this together with one other component, the probability of success, gets at, in a sense, this overall productivity. And this slide shows that the success rates and probabilities of moving forward from one development phase to the next, from early clinical development phase 1 through phase 2 testing and the large clinical trials in phase 3, and then on to product approval. That Office of Health Economics recent report compiled past study results to compare with its own analysis of probabilities at each of these stages, and they found a 44% probability of moving past phase 1, a 31% probability of moving past phase 2. That's where a lot of drop-off occurs in all of these studies. 63% in phase 3, that's the most expensive, typically large-scale clinical trial testing, which is quite costly, leading to an overall probability of success of around 7%. Now, some other numbers are a little bit higher. On the right side of this slide, another study by Damasi, which broke down success rates by therapeutic areas, again showing a lot of variation off of a fairly low average rate of success. So not surprisingly, there's been a lot of attention in recent years to try to find ways of increasing the probability of success or increasing the probability of failure occurring early, not later in stage 2 or stage 3 after a lot of time and cost of investment. One set of approaches to try to address this involves pre-competitive collaborations that often have the explicit goal of reducing the cost and time of development and supporting new approaches to the drug development process. For example, some of these pre-competitive collaborations are dedicated to getting to standard approaches for everything from data sharing to the infrastructure for clinical trials. A presentation to the Institute of Medicine in 2010 by Altshuler Gray detailed some of these kinds of partnerships, including industry collaborations to explore treatments in cancer and HIV, and industry-academia collaborations as well, such as the Biomarkers Consortium, the Coalition Against Major Diseases, which has worked on Alzheimer's disease and other neurologic conditions, the Clinical Data Interchange Standards Consortium, or CDISC, Sage Bionetworks, and others. The recently formed Transcelerate Biopharma is another new initiative in this space of pre-competitive collaboration. It's a joint effort between many pharmaceutical companies to address key development challenges, and we'll be hearing more about that example a little bit later this morning. Also, as an example of ways to try to reduce the costs and increase the predictability and efficiency of the development process is the use of biomarkers, which allow drug developers to help target their treatments and also to gain earlier insights into the effects of treatments on in terms of side effects or beneficial effects for patients. There has been a big acceleration in recent years in the use of biomarkers with more studies using them as they've been validated in the clinical setting and more use of targeted clinical trial designs as a result. In fact, this emergence of targeted trial designs as part of large-scale clinical study infrastructures has been another marked trend in recent years. For example, the so-called BATTLE and I-SPY 2 trials of the past decade have represented ways in which investigators have used biomarkers, predictors of patient-specific responses to treatment, and a rolling set of compounds being tested in different treatment arms or combinations of treatments in different treatment arms in an ongoing clinical study infrastructure to learn more quickly about which treatments are beneficial in particular subgroups of patients. There have also been efforts like by the Friends of Cancer Research to develop master protocol trials, for example, for lung cancer, to create a centralized infrastructure and a predictable model in which developers of new treatments can take more of a plug-and-play approach to drug development. Investigators can include their product in an ongoing multi-arm phase 3 trial that is continuously collecting data on control patients using standard methods and continuously updating the different treatment alternatives based on the latest evidence and a pre-specified set of rules and standards to develop more quickly the insights about which treatments are working in which patients. So these are initiatives that have been intended to reduce the administrative burden and overhead of trials and also to reduce the overall cost of trial conduct by having an ongoing large-scale clinical trial infrastructure. There have been academic efforts to improve the clinical trial process as well, such as Duke's Clinical Trial Transformation Initiative, MIT's New Drug Development Paradigm Program. These are examples of dedicated teams of researchers, clinicians working to improve methodology, data standards and infrastructure, the use of downstream registries and the like. So there is a lot going on in terms of trying to address the productivity of the biomedical innovation process, and we're going to focus on some of these issues during our time together today. In our first session, we're going to discuss the roles of a number of agencies and supporters of building a robust data infrastructure to support better scientific evidence development, better capacity for evidence development on alternative treatments. Our second panel is then going to look in more detail into some of the innovative work being done by stakeholders who are participating in the biomedical research and development process to use larger scale, more complex and innovative data sources to improve care. And then finally, we're going to turn back to some of the metrics that I discussed this morning, and also some emerging metrics of progress in related to progress in biomedical innovation, to understand what they're telling us and what more could be done to better gain insights into how to improve the efficiency of the biomedical research and development process. So that's an overview of what we're going to do today, and I'd like to get started right now with our first panel. So we ask our panelists to come on up. We're joined by Janet Woodcock, and there are more detailed biographic information in your materials, but this group doesn't need much of an introduction. You go ahead and have a seat across the panel. Dr. Janet Woodcock is the director of the Center for Drug Evaluation and Research at the Food and Drug Administration. Dr. Kathy Hudson's the deputy director for Science Outreach and Policy at the National Institutes of Health, and Dr. Brian Luce is the new chief science officer at the Patient-Centered Outcomes Research Institute. It's been, what, 2 weeks now or thereabouts?
Seems like.
Each of our panelists is going to talk about some of the new steps that their organizations are taking or supporting or encouraging to try to promote the development of better data related to evidence on biomedical innovation, safety and effectiveness of medical products that can be used to improve patient care. Then we're going to have some discussion about these issues and about the best ways to support developing an even stronger and more efficient infrastructure for developing evidence on for biomedical research and development, and that will include hopefully some participation from you all as well. So let me begin the discussion by turning to Janet, and Janet, I think there's a slide up here that you're going to use as well.
All right, well, obviously the FDA has a big stake in evidence development. The delays that you've heard about, or the long time required to develop a drug, the high cost and so forth, is greatly related to how difficult or hard it is to get the studies done, particularly the clinical studies, which are most of the cost. So we have been trying to promote more robust infrastructure, both in the development space, which would be clinical trials, as well as the post-market space, which would be surveillance of products after they're out on the market. And although conceptually these things may seem quite different at first, they're very much related. And so what Mark talked about, say the OSBA trial, what's trying to be done with lung cancer and in many other areas, I think is very important to the future success of the enterprise and being able to use data in a much more robust manner. And that is to set up standing trials, standing networks of investigators who can quickly pick up a developing therapy, plug it in to an ongoing trial that has standardized data, standardized fields, standardized methodology, and so forth, so that it can be rapidly evaluated without much of the lag. The 2-year lag, perhaps, to initiating a trial is you have to get thought leaders together, and then you gather them all together, and then you write a protocol, and then you try to round up a bunch of investigators, and then you have to sign agreements and contracts with folks, and there's this extremely long process, and that's for even a single trial. So to set up standing trials where for larger diseases we can rapidly screen candidate compounds and see whether they're effective or not, which is the main thing you're interested in initially, would be extremely, could compress this tremendously. And it is very much related to data standards, standardized data collection methodology, standardized ways of treating the data. I will add that the EMA and of course many other parties are interested in availability of data from clinical trials. This is another thing that could help speed development and inform future development better. For example, the CAMD project that Mark talked about, the Alzheimer's, the placebo data from I think 6,000 patients was pooled, and they were able to do a huge amount of analysis determining, how fast does the disease progress in these people? If you pick this outcome measure, how long would you have to wait till a significant number of people got it? And this was just from looking at the placebo group. And so now they have a disease model that can be used to simulate new trials, even in early Alzheimer's disease, and can improve the probability of success. So hopefully we will move toward, let's not reinvent the wheel with every trial and take several years to get it up and running. Let's have an efficient trial infrastructure in the United States and actually worldwide that can conduct these trials and rapidly evaluate the new discoveries. Now, on the post-market side, though, of course, everyone wants to look at healthcare and they want to analyze the data from healthcare and find out, how's it going? So we approve these wonderful new drugs, potentially, or maybe they're causing a lot of problems. Or maybe they're simple interventions like washing people with an already approved chlorhexidine soap, right, that can actually markedly improve outcomes in patients, and we just didn't know it because we had never had an efficient way to evaluate it. So what I show here, the FDA has set up something using the currently available electronic health data, which is primarily claims data, but increasingly we have the capacity as it becomes available to include electronic health record data into what we call a mini Sentinel system. And that's to look at drug safety and medical product safety post-market. That is a pilot, but it has over 120 million lives in it, and so it has significant robustness and to find what are relatively usually small signals compared to, there are not that many people in any given drug, and then not very few of them would have a significant side effect. But you still want to find out about that nevertheless. So in many Sentinel, we can set up analyses rapidly because we use a common data model. So this slide that everybody's been looking at is we should do the same that we're trying to do with clinical trials in the post-market period. So whether people are seeking to do analysis for comparative effectiveness research, or medical product safety, or they want to find best healthcare practices or quality analysis, the data that are used are very similar across all the healthcare systems. And so should we really reinvent the wheel every time we want to do one of these studies? Should we gather up investigators, identify data sources, pull them together, and so forth? Or can we build a network of data sources, what we call data partners, the people who actually manage the data on behalf of their patients? Can we build that network linked by some common protocols for data standards and so forth, and then use that data pool for any given study that we would like, depending on, of course, the permission of the data owners for any given study that would be done. So this is the vision that we have. We have built this for Sentinel, but it only is for drug safety, medical product safety. But we could see these same data could be used in multiple ways. And this system is very fast. We can query our contractor, Harvard Pilgrim, and get an answer back on a simple thing in a week, or even faster. And we can get complicated questions answered actually very rapidly as well. We've cut years off what we used to do when we were investigating a safety signal by having this system available. So wouldn't that be very helpful for all the kind of evidence that we'd like to gather about healthcare? So that those standardization, infrastructure, that's where we see the big payoffs coming in both drug development and healthcare safety.
Great. Thanks very much, Janet. Kathy?
Great. So I'm going to go a little earlier in the pipeline and talk a little bit about basic research and the contribution that NIH makes to advancing basic research and really providing some of the knowledge and insights and targets that are then translated, hopefully, into new drugs and therapeutics. And of course, the part of the reason why we see such a high failure rate, which Mark talked about, is because what we're starting with in clinical trials are the wrong targets and the wrong compounds. And so if we could do that in a more efficient way, identifying those targets and those compounds, hopefully we could increase our success rate and decrease the cost. The title of our little session is Creating Novel Sources and Applications of Data Through Policy. And so I wanted to talk about 3 sources of new data and then 3 sort of policy innovations, if you will, to try to maximize the use and utility of data. So the first source of data, new source of data that I want to mention is a new initiative that was announced by the President in April, the BRAIN Initiative. And the focus of the BRAIN Initiative is really a basic research undertaking to try to understand how the 100 billion neurons and the 100 trillion connections between those neurons function in a normal state to control things like motion and memory and speech, but also in brain disorders and diseases. And of course, the toll of brain disorders and diseases on the economy and on us personally and our families is enormous. Recently, the number of failures in neuroactive compounds in clinical trials has been abysmal. And so anything that we can do to improve the basic understanding of how the brain works when it works well and how the brain doesn't work in disease or disorders or trauma would be, of course, tremendously exciting. In this economy, NIH is faced with the sequester. We lost $1.57 billion overnight. We're going to fund 700 fewer research grants this year than last year. And so in that environment, being able to collaborate with others and leverage each other's know-how and expertise is really critical. And certainly we're collaborating with PCORI and with FDA and with many others. And on the BRAIN Initiative, we're collaborating with the Defense Advanced Research Projects Agency, which is able to move swiftly and nimbly where sometimes we don't. And so we're learning from them. And also partnering with a number of private foundations and hopefully with companies. A lot of the focus of the BRAIN Initiative is going to be on developing new technologies. And so this arena might be ripe for being able to use a relatively new ability that we have to give out prizes. So you can imagine holding out a prize and saying, if you can develop the gizmo that will do various kinds of detections in the brain of brain activity, there will be a prize awarded. So we're excited about that. We're just at the very beginning of planning that initiative. The second collaboration that I want to mention is the Target Validation Consortium. As Mark mentioned and Janet mentioned, the high attrition rate is of significant concern. And the question that Dr. Collins, the director of the NIH, wanted to address is whether there were ways in which the NIH could work with pharmaceutical companies and others to try to sift through the tsunami of potential targets in order to identify those that would be more promising. And so we have been working over the last, I guess, year and a half to try to map out some pre-competitive areas in which we can collaborate together and are very close now to seeing this come to fruition. The activity is going to focus on 4 different disease areas as sort of a pilot to see how well this playing in the sandbox together works. The first is Alzheimer's disease, which of course is a presidential priority and of high importance, and of course with new guidance from FDA about how to be able to conduct those trials. Hopefully that will be moving more swiftly. The second is in diabetes, the third is rheumatoid arthritis, and the fourth is in schizophrenia. So we're excited about that collaboration being able to work together, facilitated by the Foundation for the NIH. The third area of data generation that I want to mention is also a collaboration that was quite fun to be able to put together and is lodged at the new center, the newest component of the NIH, which is the National Center for Advancing Translational Sciences, or NCATS. And this involved trying to pre-negotiate arrangements between ourselves and universities, and between universities and companies, and between ourselves and companies, in order to invite companies to provide compounds that that were abandoned, to provide those compounds to the biomedical research community, and basically repurpose those drugs through a three-way collaboration. And those awards were, the first round of those awards was just announced last month, with 9 grants being awarded, and interestingly, with a substantial proportion of those being for compounds that pass the blood-brain barrier and have demonstrated safety in humans. So this is another novel approach where, by working together, we might be able to attack some thorny problems that otherwise would be hard to get to. So those are some of the data generation activities that we are working on. I want to turn just for a second to a couple of policy issues. We have really been struggling at NIH with the issue of the clinical trials that we support and how to make sure that we fund high quality clinical trials that are managed well from the NIH, and that the data and results are reported so that we don't repeat the same mistakes, so that we don't fund the same trials over and over again, and so that we can learn from the cumulative data. And so we have been working with FDA to try to put out the guidances for ensuring that all folks who do clinical trials submit their data, submit their results into clinicaltrials.gov. We're trying to improve that process as we go. The last policy reform I'll mention is that the administration a year and a half ago put out a proposal to reform the regulations that govern the protection of human research subjects in research. And of course, anyone who has done human subjects research knows that sometimes those rules are more burdensome than they are of benefit to the patients in the trials. And so we are anxious and eager to put out the reforms to the common rule in order to facilitate research and increase the protections for human beings who help us so much in research. And with that, I'll end. And
Okay, thanks. Looks like you want to come up here, Brian. Thank you.
Thank you, Mark. This is a real pleasure to be here. So I can just continue. Yeah, great. So I'm going to work off some slides, in part because I'm so new. I'm learning as you are, so I'll be interested in what I have to say. I'm as the new Chief Science Officer, I'm really pleased to be part of PCORI. You all probably know at this stage that PCORI is an independent, not-for-profit research institute authorized by the Patient Protection and Affordable Care Act. It funds comparative effectiveness research, providing for patients and those who care for them with the information that they can use to better inform healthcare decisions. Well before I joined PCORI in July of 2012, PCORI sponsored a national workshop to, in essence, to get a footprint into the use of electronic data, and we are now in the process of putting that footprint to work. That includes clinical data networks and a number of patient-powered research networks. We are going to be funding up to 8 clinical data research networks, which are system-based networks such as care delivery systems and health plans, with the potential to be expanded into comprehensively covered cohorts of over 1 million persons, and up to 18 patient-powered research networks, which should consist of groups of patients with single or a few related conditions organized to participate in research. And finally, we'll be funding a coordinating center to pull all of this together. So this is sort of a graphic, which is sort of gives you a sense of how it's supposed to work. It doesn't help too much as to how it's going to work. We'll have a steering committee that'll include not only the awardees and PCORI, but also the various agencies you see here, and we'll have a scientific advisory board and a special expert group as well. Give you a sense of the scope of what we're planning on doing. We've allocated $56 million to support these 8 different existing CDRNs for the first 18 months, which is really the initial phase to get their act together. These will be cooperative agreement awards. We're expecting we're requiring actually at least 2 healthcare systems to be engaged with a willingness and capacity to work together for standardization and with other awardees. And a lot of the issues that we're talking about in terms of standardization is key, and I have no idea of how we're talking about standardization with Sentinel and so forth, but obviously that's a key issue. And a willingness to participate and collaborate studies in terms of data sharing and national research infrastructure. In 18 months, we're hoping and expecting that, requiring that there'll be a million patients enrolled in each one of these networks with the data standardization that we just talked about, with patients and the system and clinicians engaged in governance and use. And importantly, and I think this is really a major challenge, but an important plan of ours is that these networks are capable of implementing clinical trials, and that's one of the requirements for our awardees. We have the letters of intent in-house. They're really exciting. We've got a lot of the major networks around the country. Probably some of you are involved with them, where we just blown away by these letters of intent, both in the clinical data research networks as well as the patient-powered research networks. So we're awarding, or allocated $12 million to support up to 18 of these patient-powered research networks, again, for the first 18 months. So obviously a lot more will follow after the 18 months, which we hope then we'll be able to link a lot of these together. Again, these will be cooperative agreements so that we will be able to work with them collaboratively. And as you can see there, patients, these agreements should have patients, at least initially, with a single condition or similar conditions, interested in research participation and ability to increase size and diversity and willingness to build standardized databases and to link with others. And we're looking for, within that 18-month window, a target size for each one of these networks, 5.5% of the U.S. population with that condition. We're particularly interested in areas of rare diseases, as you see. Importantly, that these patient-powered networks are able to collect patient-reported data. As you may or may not know, PCORI is extremely focused on patient data that's and outcomes that are important to patients, and that certainly includes patient-reported outcomes. Patients will be involved in governance, as it's true also in the clinical data networks, and so forth. So that's all supposed to work together. There are lots of challenges. One of the things, interestingly, as I was looking through this morning, is the challenges of the IRB and legal issues, especially when we're talking about mounting clinical trials within this. And the whole point of pulling this together is to, of course, we're talking about post-market if we're talking about drugs, but we're hoping and planning that this will work seamlessly in terms of both observational research and experimental research. Of course, all of the various actors will be part of the system, and some sort of a network. And we imagine a new world, in essence, to be able to have a dynamically linked multiple patient and clinical data networks, allowing for mounting learning randomized trials, such as adaptive, Bayesian kind of trials, whether they're definitive or not, but breaking into observational studies and back and forth, so that as you, for instance, do an observational study, and you have hypotheses that need to be tested, we could break into clinical trials. This is the imagination here, and all guided by clinicians and researchers, patients, and stakeholders. So it's a major endeavor that we're going into. Thank you very much.
Thanks very much, Brian. I'd like to get the discussion going around the large amount of information initiatives that you all presented. And I know it's very hard to summarize such a big scope of activities, but it did seem like the efforts that you all are collectively undertaking hit on every aspect of the innovation process, starting with the translation of basic science into better models, better knowledge that can be used to target actual clinical trials early, and a more efficient late-stage design as well, standardization of the data and the best protocols and the patient protection steps for conducting these trials and other ways of generating evidence, and then an ongoing infrastructure, what Brian called a new world of dynamic real evidence generation from data, including much more data from patients through adaptive trial designs and kind of a large ongoing infrastructure for developing evidence. Now that's a great vision if you put it all together, but we're clearly a good ways from getting there. And in discussing the obstacles, I'd like to go through each of these three maybe quickly, or we'll see how far we get in the next few minutes, and get a sense from you all of the extent to which you think the challenges are related to resources. Kathy and others have mentioned, and I talked about kind of the leveling off of a lot of the input, the funding going into these activities, not just on the NIH side, but on the industry and further downstream side as well. And also things that can be done to make this process more efficient. Are there policy changes building on some of the standards that have been developed and some of the initiatives that you all are undertaking? So maybe starting on the Kathy, your area, and translating developing more basic science, translating that into much more confident guidance as to where the clinical studies should be conducted, what targets, what patient populations. Biggest obstacle there is resources. Are there ways to do this more efficiently?
Well, certainly resources are an issue, and I mentioned the effect of sequester this year, but of course the sequester is anticipated to last for a long time unless there's a miracle in sight. So we're looking at $18 billion in lost resources, and so that really is an incentive for developing new ways of being able to do this better. I think linking up the basic research enterprise to focus on clearly identified problems in the translation pathway is one way where we can really help overcome some of these challenges. We have great basic researchers out there, but they're not necessarily connected to solving a specific problem in clinical trial design or in developing new molecules. So I think part of it is providing that focus. We vary from institute to institute at NIH of how much of the research that comes in the door, applications that come in the door, are solicited. So in hard budget times, it might make sense for us to be much more targeted in asking the research community to focus on specific problems that we can work with industry and FDA and others to identify what are those problems that our community can solve, and having maybe a little bit less investigator-initiated work and more targeted work to try to overcome these problems.
I judge by the brain initiative and by some of the other targeted areas you mentioned around diabetes, rheumatoid arthritis, schizophrenia. Those are where you're thinking the best opportunities to do this are?
Well, those four areas were really identified by the the steering committee of industry, academia, and NIH working together as areas where there were specific opportunities and, frankly, specific business interests from the companies that are involved.
Janet?
Well, I think one of the problems that's faced in the early development enterprise is that it is well agreed upon, and I have been corrected on this by the cancer center directors, okay, and as far as the numbers, okay, that about 3/4 of the published literature on pathways cannot be reproduced.
Did you have to bring that up?
Yes, okay, because if you're talking about inefficiencies, all right, so the standardization process and the developing standards needs to move back.
Yep.
Okay.
That's right.
Preclinical.
And
Right.
this is
That's
not
right.
about like fraud or anything. This is about standardized reagents, standardized methodologies,
Underpowered
repeating
studies.
experiments, not sending in the best result and leaving the others behind. And it is a big problem because if you identify a pathway and start developing molecules against the pathway and it actually turns out
Right.
not to be that operative in the disease of interest, and you spend several $100 million developing a suite of molecules against that target. What the pharmaceutical industry does now is try to reproduce all these published experiments in their laboratories before they start spending the money. And of course, that's where this data have come from, that they can't reproduce it. Even if they talk to the investigator, they get the same reagents. They try to do everything the same, and it doesn't happen. So that's a problem that, in my mind, on the discovery side, is a really big problem. Unless we can understand basic biology and have some rigor in our understanding, it's complicated enough. Biology without having all this false information in it.
Can I respond to that quickly? So this is a huge problem. There have been a number of cases, specifically in the Neurology Institute, where experiments, mostly in rodents, mice and rats, that have published in single-word journals, Nature, Science, Cell, and have built careers where they have specifically gone out and paid for other laboratories to try to reproduce those findings, and the overwhelming majority have been not reproduced. So what's the problem there? And we've been looking at this very carefully. Harold Varmus and Story Landis at NIH have really been leading the charge for us in terms of what do we need to do at the review stage. So when those applications come in and people are proposing these experiments, whether it's in C. elegans or yeast or mice or monkeys, how do we detect early that the outcome of that research is not going to be sufficiently robust and rigorous to be able to, for anybody to be able to invest in?
Be actionable, I
Yeah,
call it.
that's right.
Be
And so
actionable,
we're developing some initiatives that we'll probably be rolling out over the course of the next several months to really make sure that we are funding rigorous, robust science and not just the stuff that wows the peer
So you see
review
that
process.
coming through the NIH funding process,
Yes,
including some
absolutely.
standards to promote reproducibility in the
That's
results
right.
that come out.
That's right.
Janet?
And then on the very next step, okay, the biomarkers, we have the same problem, okay? Thousands of people develop biomarkers, they publish them in journals, they make their careers on them, and we have no evidence about their action, about their performance, yeah. So I know Ann Barker out in Arizona and some other groups are trying to start looking at, can we put in place some standards there? Again, this is more in the basic science area. So how do you decide that a biomarker has some reliability and robustness as far as its evidence base?
And how far are we along on standards and reproducibility there? There are a number of biomarkers that have been accepted in recent years by FDA, other regulatory agencies, and are being used in
Yes, but
clinical
they're
trials.
usually like geriatric and they've been around since the 70s, some of them. I mean, some of the genome
grandfathers.
sequencing is it's not that reliable, but if you do it enough times and it isn't that hard, then you can, and it's not that expensive, yeah, and it can be done by many hands, so in many laboratories. So the sequencing information is of a different character because it's not about a dynamic thing, it's about what's in the genome, so to speak, at least the exome. So so that's when we've been accepting some of those biomarkers is true, you know. However, much of the other biomarkers that are more dynamic that you'd really be interested in a disease, okay, that's why there's so much controversy about this, about diagnostics and so forth, because the evidence base usually isn't there. Look at PSA. That's the one I like to use. We don't use that for as a biomarker in prostate cancer. It's been around forever. It's used as a screening tool. That's becoming more and more controversial.
Right. We've talked, this is great stuff. I do want to move to, we've talked a good bit about the preclinical work turning into models that tell us what to study in which patients and what outcomes. I do want to talk briefly about the clinical study networks and evidence themselves. And you all described some work that's being done to standardize methods to generate, as Brian said, a ongoing network capacity at a large scale. But we still have a long way to go there, it sounds like. Further thoughts?
Yeah, well, I would just like to say my usual controversial self. Okay, I think the idea of letting 1,000 flowers bloom in the clinical space will result in the same problem that we have in the discovery space, in the publication, I like the single word journal space, okay, which is, okay, if you don't introduce rigor and standards into how you describe a phenotype, you might as well just throw the baby out with the bathwater. You can have tons of data. That's what we showed in OMOP, okay. You can have huge amounts of data and you can be reproducibly wrong. You can be wrong in this data set and be wrong in that data set and be wrong again. And often you're wrong the same way, and it really can mislead people. So rigor, I think, needs to be a watchword, and that's not something people like. They think we can just throw all these clinical data together and voila, we'll learn the truth.
And are there reasons for confidence that these differences across systems and data collection and differences perhaps in the electronic methods of generating the data themselves can be overcome? Is that what Sentinel seems to be saying,
yes,
at least in some areas?
Well, that's claims data primarily, don't forget, and that's a special beast with its own special problems, as Brian knows very well. However, when you're talking about the electronic health record, what I hear from my clinical colleagues right now, it constrains you, depending on the record you're using, to certain ways of portraying information, okay? And so, but yes, Sentinel has showed us you can correct for these things. There are ways around this, but you can't just link everything up and then cross your fingers and hope for the best.
Right. And Brian, you're going to be trying to do this with the clinical data research networks that you were describing as well. And I know you've been only at PCORI for a short time. You've been working in this field for quite a while. Are there reasons for optimism about being able to combine electronic clinical records, not claims data, in ways that will allow for consistent, reproducible, accurate evidence development?
Well, I mean, there's obviously a long, long way to go, and it's a lot messier world than, preclinical world where you can control. I mean, if you have problems controlling everything, because you can't control everything in the real world, and part of our interest is studying the uncontrolled real world, to be honest.
Right. Right.
And so everything we're talking about is, a bigger problem in sort of spades in terms of variability and standardization and so forth. Not necessarily standardization in terms of the data itself. I think the major issues that we're going to face is in part, like I mentioned, the IRB and legal issues associated with being able to move quickly in real-world settings, especially to the extent that we're talking about randomizing patients to resolve specific issues. There are other structural, cultural, analytical issues that I am particularly interested in addressing, and that is the observational data analytical world and training and infrastructure and experts and researchers with the experimental world, because those have to come together. If we're going to be able to seamlessly generate hypotheses and then spin into a trial to resolve specific issues within real-world settings, we have to somehow come to an agreement about standards of evidence, how much is enough, the tolerance for uncertainty, which is going to be much different, I think, than in some of the things you're talking about here. So there are a number of issues that are going to be extremely interesting and quite exciting, I think, that I don't think have been dealt with hardly at all in order to get from here to there.
And on top of that, you're trying to bring in a lot more data from patients where...
Well, yeah, I mean, then we have the entire dimension of bringing in patients into the whole process about what's important, what are the questions that are important, what are the outcomes we want to study, and then the governance and so forth. And of course, that's a trick all by itself, and we're learning a great deal. And Macquarie's actually learned a great deal over the last couple of years of how to bring patients into the research process itself. But that throws in more uncertainty and challenges in and of itself.
And has that provided some insights? All of you mentioned in one way or another the IRB process and providing adequate and effective patient protections and research while still encouraging and engaging patients. It sounds like things are moving on that front with revisions in the common rule coming, with some models out there and some of these larger trial networks like City and I-SPY 2 that may help address these issues. Any further comments on this topic?
Well, I think some of the things we're looking at, I know NIH is because obviously the trial of washing people and at cluster randomization within the standard of care, okay?
Huge
and
issue.
You can do that and then capture, what is that? Okay, first of all, instead of randomizing people, if you don't know what the standard of care should be, and everybody accepts different methods, and that's studying quality of care or studying care in general, you can randomize hospitals rather than randomize, or units or something like that, rather than randomizing people, not have to do the informed consent process, which adds a huge amount of burden and lack of sort of real world-ishness to the whole enterprise. So you can do that, and then you can learn. What we have learned is you can learn really important things by doing this, and so we're exploring that. I know NIH is continuing to explore that. So that's one way that I think we can learn. And there are natural experiments that go on sometimes for drugs where a formulary won't cover a drug, and so you have one area that doesn't have it and another that does. And if you have electronic capture of data, and we also, Mark, we've been doing, we started patient-centered drug development under the 5th PDUFA, and although the sequestration has slowed us down, we are working, we've had a number of meetings with patients and working to get them to generate instruments. But again, this isn't just random experiences. They're going to generate an instrument where they can record patient-reported outcomes of what they care about. And we've learned about that. And we've had some patient groups come in with their trade-offs. Like, how much of this would you give for this much benefit? How much risk would you tolerate? And that's very informative, I think, for drug development. And that's, I think, the direction of the future. So whether the databases themselves and using them is the best way to do that, or some more overt, like the patient-powered research, where you actually go out and ask people, elicit this, and develop an understanding of both the burden of disease from the patient's point of view and what the trade-offs they tolerate in terms of a therapy that's very interesting, and we're moving ahead with that.
Can I say a word about IRBs? So in the proposal that the administration put forward a while back, was a requirement that for multi-site domestic studies there be a single IRB. And we know that when there's a single IRB that the whole review process works faster and more efficiently and with less frustration for the investigators. When I was at Hopkins down the street and doing some human subjects research, I was collaborating with 5 other universities, and we had to go through their 5 IRBs plus my IRB plus a contractor's IRB, and they were line editing a survey instrument.
Right.
That was not useful to anybody, right? So we have already done a lot of experiments with single IRBs. So the cooperative oncology groups now require that you use use the NCI IRB unless you can prove that you can turn it around faster than the central IRB. And so far, I think no one has succeeded in doing that. The National Children's Study has a single IRB. We're experimenting with single IRBs in NeuroNext, which is a network which you emphasize. Let's build these things once and then redeploy them rather than setting them up, doing a trial, taking them down, repeat, right? It's not an efficient way to do business. The other innovation, I think, in the human subject side is being able to really engage patients in the process of being partners. So we have for way too long had a policy in place where once we take your specimen, if we rip off your HIPAA identifiers, we just remove the label, we can do whatever we like with those specimens. But of course that limits us, because then you can't go back to recontact to do deep phenotyping. You're limited with what you can do. What the new proposal recommends or proposes is that investigators or clinicians at the get-go engage with patients to say, are you comfortable being a part of this research enterprise? Not being specific about every little possible experiment that might be done using data or specimens, but asking people to participate as partners. And that's something we can do some research on as a part of your network.
And we will.
Yeah.
That is part of our remit, in essence, to do.
So
I think if you bring the patients in early and they're actually part of the governance and so forth, then you are more likely to get buy-in across the board.
Right.
Sounds like this infrastructure being developed is going to have a lot more active role for patients. I would like to open this up to a few questions or comments from the audience. If you could raise your hand, we'll get a microphone to you as soon as we can. So one up here and one in the back.
Hi, I'm Eric Kaplan. I have biopractices. I have a question. As I was listening to your conversation, I was just thinking about this. I think virtually every major medical breakthrough has either been the product of insight or serendipity. And I was asking myself this question, has there ever been a significant medical breakthrough that any of you can recall, I can't think of any, that have been the product of rigorous analysis of assembling massive amounts of data and then analyzing it? In other words, what's really the payoff for this in terms of innovation? I see the payoff is in terms of what we should pay for, maybe, to ask the question, does it work? But in terms of real innovation, making a difference, moving the needle, can any of you think of any example involving the massive assemblage of data and analysis? Thanks.
We don't have that much experience with the massive assemblage of data because we haven't done it yet. So we have launched a new project called Big Data to Knowledge, where we're basically going to be supporting centers of excellence to be able to do experiments in how to assemble data and then how to develop the tools and methodologies to be able to manipulate, mine, and get knowledge out of that data. But we don't have that many examples of where there is aggregated real big data in biology. In physics, they've done it. In other domains of science, they've done it. And I don't know anything about those areas of science. So I'm sure that we should learn from them about the insights that you can gain from big data. But we haven't yet really had a home run from big data because we haven't yet developed the tools and technologies to mine it.
Now, there is kind of an indirect aspect of big data, I guess, which is that in developing these larger research and clinical trial networks with standardized methods and protocols, you're in effect able to combine data from many more patients and generate the kinds of evidence maybe that have gone into previous studies in subgroups of patients more efficiently. And that seems to be having an impact in I-SPY 2 and many of these other trial networks.
Yeah, well, those data are to some extent structured, and the problem is what I'm objecting to too, I think, is that the belief that we just get unstructured data and we have tremendous insights. That has to be seen as a hypothesis in biology, not a necessarily objective, because biology is many logs more complicated than, say, particle physics. All right, so I think though that, that, things like the National Cancer Genome Atlas and things like those are things where there's something, actionable that you can find down in the cancer genome, and you can generate hypotheses and targets and grow from that. And that's probably one of the better examples. But there, we've spent 30 years understanding cancer pathogenesis, and so we're not there yet, but we know a fair amount about it. And so those can yield targets now, and those targets are, some of them out on the market and are the treatments for those targets and are effective. So that's kind of a mini example, I would say.
Great. Other questions in the back? See two more.
Hi, my name is Rick Blake. I'm with Strategic Health Resources. We represent the small biotech industry, a lot of them. And I find it kind of curious that we're talking about biomedical innovation without small biotech at the table. And I mean, I'm not talking about big pharma. This is sort of like big government talks to big pharma. And I think really we need some regulatory changes both in the FDA and at NIH to be able to have some of the drop-off in the small biotech sector stop. I mean, many of these companies are going out of business while we're talking about data this and data that. I'm not saying it's not necessary. I'm just saying the investment climate in the small biotech industry is horrible. So that while you're talking, probably 4 companies have gone out of business. So one is I'd like to alert you to the fact that Danny K. Davis has the Small Biotech Protection Act, which he could have been here to talk about, I suppose, and which has some of the regulatory changes we need at a federal level, and also a budget-neutral solution for getting the investment climate back for the small biotech firms who are the real innovators in this sector.
Okay, we are going to come back to these topics a little bit later, but I would like to ask the panel, from the standpoint of a small startup, maybe venture-funded company, what are the implications of this movement towards more data standardization, larger clinical trial networks? Is that helpful to them? Is it irrelevant?
Well, first I'd like to say every year we see an increasing proportion of the people who make those new molecular entities that get approved that Mark showed. Increasing proportion of them are small, it's the first product they've launched, okay? So they're small entities. I think for the FDA, probably the most profound change lately for small biotech would be the breakthrough therapy designation and the fallout from that, the kind of things that can be done because that can help streamline development. I believe that if we had standing networks that companies could plug their promising products into, it would tremendously decrease their costs and time.
Is that happening now for smaller biotech companies, or do they still feel like they have to kind of do this all themselves, but now they've got more standards and so forth that they have to master first?
There aren't standing trial networks, with the exception of the very few that you've mentioned, that could take a product and do, say, there's nothing right now that could do a registration trial for a product, for example, except maybe cystic fibrosis, multiple myeloma, those patient groups, okay, those foundations, they have put in place the infrastructure to get that done, and they've had multiple products approved. Some of them.
Some good successes from using those patient-supported or patient group-supported
yeah, so
trial networks.
that's a great model for the smaller diseases. For the large public health-related diseases, we need larger infrastructure. There's so many patients out there, and they don't get enrolled in trials because there aren't any trials, that are out in their communities.
The patient-powered networks may be a foundation for some of this, I wonder.
Yeah.
We'll have to see.
We have maybe time for one quick comment question. Ellen, did you?
Yeah. Ellen Siegel, Friends of Cancer Research. So Sunday there was a very provocative op-ed in the New York Times from Clifton Leaf addressing clinical trials and addressing the fact that the problem we have is not money in the system, but we're doing the wrong work. So, and he's going to be writing a book, and it's going to be very, very provocative, and there's much in it that we'll agree with, much we will not agree with, and I think it may do harm. However, specifically, I'd like to ask the question about clinical trials, because we are doing big trials, and often we're not doing good trials, and we're not dealing with the heterogeneity of patient populations were not asking the right questions. And this is all of us. This is, you know, the cooperative groups, this is companies. And what has to change about the way we conduct trials? A lot of companies, or a lot of even smaller biotechs, or frankly even the NIH, are conducting trials that they think the FDA wants, or they're collecting data that they think the agency wants, and often it's not necessary, and they're asking the wrong questions. How are we going to get to a better point where we're doing this more efficiently and asking questions that are actually going to work? Because there is, we can all agree, a lot of money in the system, and we can all agree we need to make some changes. So how are we going to get the memo out to do smarter and better trials?
Well, I would ask, you know, where we have departments of biochemistry and molecular biology and genetics in every stand-up university all around the country. Okay, where are the departments of clinical trials? We spend probably hundreds of billions of dollars on clinical trials. We don't have the right experts. And I would disagree with Kathy. I don't think taking people from the basic sciences and trying to get them to figure out how to do clinical trials is the right answer.
That's not what I really suggest.
Okay, I figured that. Okay, so basically, I don't think, I've always said, and people take issue with this because we spend so much money on them, but doing clinical trials is a cottage industry. And to leave it in the hands of the pharmaceutical industry, their objective is not to improve the clinical trial enterprise. Their objective is to get their product evaluated properly, okay? And so there hasn't been a support for the scholarly, the academic, the actual rigor of how you would do this right. And that's why we have what I guess we get what we pay for. So I think that the rise of
promising steps to address that.
Trial networks are supported, okay, and I think Transcelerate is looking at some of this. I think that helping to support investigators, train sites better, do things like that. But it's if we have standing trial networks that are supported, you develop a class of professional investigators, okay, and they become experts. That's what we need. We have like 10 experts, right? We need lots of experts in every disease who make it their business, their life's business, to evaluate medical treatments or interventions of all kinds. So I know I'm talking a lot, Mark, but I really think that would be what would make the difference.
Well, and we do make investments in clinical trials. It's not a department, but with the biggest single program at the National Institutes of Health is the CTSA, it's the Clinical and Translation Science Awards. And we have been investing in them since 2006, 2008. And we need to now turn to them and have some expectations about how they're going to help us improve this process. So it's $460 million a year that go to the highest, best medical research centers in the country, and we now need to turn to them and say, you need to be our partners in solving these problems.
I'd like to thank the panel for an excellent and wide-ranging discussion of the challenges and also the policy steps to address improving innovation.
And I'd also like to move right into our next panel. If our second panelist can come up to the stage, I'll introduce them as they are doing so. Again, there are more detailed biographies on all of our participants in the materials that you have in your packets. Now we're going to turn to some leaders of organizations that are undertaking some of this work that we've been talking about so far today, some of this work on developing better evidence, including using novel data sources and integrating complex data more effectively from a broader range of sources, including new pre-competitive collaborations and otherwise. The goal of many of these efforts is to improve the innovation process and also to improve care delivery. I'm very pleased to be joined by Dalver Gill, who's the Chief Executive Officer at Transcelerate Pharma, which has already been mentioned this morning. Jeff Trent, the President and Research Director of the Translational Genomics Research Institute. Dr. Martin Cohn, the Chief Medical Scientist for Care Delivery Systems at the Watson Research Center at IBM. and Gabriel Eichler, the General Manager of Client Services and Strategic Initiatives at Patients Like Me, one of those leading patient-powered research and evidence development networks that we talked about before. So I'm going to ask for all of these panelists to make some opening comments to get our discussion going. Dr. Gill, if you don't mind, why don't we start with you?
Thank you, Mark. Just to introduce myself, Dalvia Gill, CEO of Transcelerate. It's been mentioned by a number of the panelists earlier. So let me just, maybe it'll be helpful to try to demystify a little bit about what Transcelerate is. I find that audiences often have a perception of what it is, so let me just kind of give you a sense of what it is. It's an organization that is less than a year old. It was incorporated in August of last year. It's a not-for-profit entity, and so its inception came about through various discussions that R&D heads from the biopharmaceutical industry have in various forums over the years. And they've always spoken about, well, what other things we need to do to improve the efficiency of the R&D process. And there are many things, as you know, often self-inflicted. However, despite all those discussions, they would go back, they'd do their day jobs, they'd come back a year later, and they would have the same problems, and they would discuss them all over again. This happened for many years, I understand. But a couple of years ago, they finally decided that the pain is no longer bearable. The circumstances and cost of developing drugs, as Mark outlined earlier, are no longer sustainable. There are some amazing molecules and elegant science that has come from various sources, including from industry, that are not being developed because it's just not cost effective for them to develop, and the return on investment for the investment they would make, it did not make sense. It was not a sustainable model. Long story short, they decided that there were some very obvious areas in the R&D process that could be tackled in a collaborative manner. I think one of the problems we have as an industry is that we look at everything as a competitive venture. There are many things we do that are really not competitive, and you could argue, and I've had a CEO of one major pharmaceutical company argue that the only real competitive thing we have is the molecule itself. And you could argue everything else could be, you could argue that it's not all that competitive. That said, looked at target validation, they looked at potentially improvement in clinical trials, in translational research, big data sharing, et cetera, various things we could have tackled. But it seemed obvious to everyone that the clinical trial space was one that needed some immediate attention. There was some obvious, glaring, low-hanging fruit, areas of inefficiency. I bet you all the people in this room who do clinical trials, you would probably pick the first 5 or 10 initiatives that Transcelerate's tackling because you would know yourself that they don't make sense the way we do them today. So a company was incorporated. It's a very lean organization. It has a board of directors. It had 10 founding members. Those are 10 of the largest pharmaceutical companies in the world, the Pfizers, the J&Js, the GSKs, of that ilk, very large companies. And then 7 more companies have joined Transcelerate since its inception, companies like Astellas and Biogen, IDEC, and companies of that nature who now make up 17, and it's probably towards the end of next year, probably around 25 to 30 based on the interest that we have out there. So a growing concern, a very flat organization, it's very lean, it's primarily funded through cash contributions, subscriptions and dues, but that's actually a small part of the contribution to Transcelerate. The primary contribution, and one of the main differentiators as to why we've been successful, and I'll talk about that in a second, is that the companies provide their expertise, and not just people sitting around with nothing better to do, but their best brains, with their problem solvers. So in this particular configuration, the companies define their own problem with their own people. They fix the problem with a solution that they come up with their people, and then they go away and implement them, or not, depending on their desires. In this particular configuration of this collaboration of Transcelerate, no company has to implement any solution if they feel it's not appropriate for them. However, they are all involved at this point in funding and looking at various things that we can tackle. So let me just quickly talk about the low-hanging fruit I described earlier in the clinical trial space. We've launched 5 projects, 4 of which essentially have started to deliver, and 1 that will deliver at some point. And some of them are going to be very obvious to you. Risk-based monitoring. People speak about that, quality by design. CTTI has been working on it. Various different organizations have been working on monitoring clinical trials more efficiently. The checking of every data point in every site in every country, it just did not make sense. And in fact, when you look at the data, it was pretty evident that systemic issues and systematic issues are harder to find in that because we were really concentrating on the minutia of the data. So in a risk-based monitoring approach, we worked with Janet's group at the FDA, we worked with the EMA. 17 companies came together and defined a methodology. None of it rocket science, none of it groundbreaking, but just a consolidation of thinking. That in itself is a major step forward. It allows the member companies and people outside of the member companies, and that's an important point I want to make, the solutions that come out of Transcelerate are often not just for Transcelerate members. They are made available to anyone who feels they want to use it, obviously. So in the risk-based monitoring paper, a position paper, toolkit was published at the end of May. It was published on our website for anybody to use. 6 different pilot studies have been launched by the member companies with the knowledge of the regulators in Europe and the FDA to take that methodology, to pilot it, to refine it for future iterations of that particular position paper. That's the first. Second one was mentioned earlier, site qualification and training. It's nonsensical in that we train sites, we train them for GCP, we get them qualified, and then along comes a totally different company a week later and they do the exact same thing. It just doesn't make sense. It's illogical. So the companies Transcelerate, and I'm making it sound simple, but getting 17 companies to agree to do anything together is actually quite a challenge. So you'll forgive me for simplifying the work of my colleagues, but we'll just take it for granted that they're eager to collaborate at this point. But 17 companies have agreed on what GCP training means and how will they measure it and how will they recognize it. So today, it's an active network. If, for example, J&J were to qualify a particular investigative site for various aspects of the ability to do a clinical trial, the other 16 members will not replicate that effort. They will take the certification and they will move forward. Now, it doesn't sound like much, but believe me, that's hundreds of millions of dollars of wasted money that we're now going to save. And more importantly, we're going to take the burden off the investigators so they can concentrate on getting patients into a clinical trial and their general patient care. That's what they're there for. The third project is a shared industry portal. Every company communicates with its investigative site through an electronic portal that they've developed that they feel is a competitive portal through which they exchange feasibility information, safety information, document exchange, etc. Well, these companies have now agreed to come up with a shared portal that they're developing together. We just had board approval of it about a month ago. And in various stages, we will release a shared portal that we are working with investigative sites on. For example, the Society of Clinical Research Sites is a newly formed trade organization for investigative sites. We're working with them to get investigator input so we can build a portal that makes sense. So study coordinators don't need to remember 50 passwords to do their day job. Again, simple in concept, but actually very challenging from a technology perspective. But we are absolutely tackling that. Comparators is another project very quickly. Today, if you're doing a comparative clinical trial, you need to source the comparator from another company. It's actually a painful process. You may go directly and have a bilateral agreement. Often, you go to a third party to get the drug. It costs you more money. There's counterfeit drug supply in the supply chain, more so now than ever before. You don't get the information you need on stability of the study, the drug, its temperature excursion information as readily as you would if you got it from your partner. So a network has now been formed. We haven't put a press release out yet, but as of Friday, we now have an active network in which member companies of Transcelerate will source each other's comparators directly to the other company and proactively tell them when they may need it. Now, this particular project is going to have knock-on investigational product, bulk supply, placebos, all the various things that are very problematic in the drug supply. And one of the most important things for patients is we will reduce the risk of counterfeit supply, drugs in the supply chain as a result of direct sourcing from the company itself. And then the last project is we are working very closely with CDISC, with FDA through a collaboration of CFAST, where we're developing efficacy data standards for various therapeutic areas. And right now we've launched a pilot in asthma and diabetes. And we hope our stated goal together with FDA and CDISC, is to do about 55 therapeutic areas in a 5-year space. And that in itself, we talked about standardization, that's what we're talking about here, so that be it diabetes or Alzheimer's or something else, we have standardization of efficacy data as we had done for some safety data in the industry and in clinical trials many years ago. Next steps, we're going to be launching a number of new projects. I can tell you some of them are very exciting, a little bit more far-reaching projects. We've tested the concept that we can collaborate. We can collaborate in this particular configuration. We can deliver. And now I think we're taking a look at, well, there are so many things we can tackle. What other things do we want to tackle? For example, a pediatric clinical trial network is very high on our list for the very reason that was described earlier, which is many of these biopharmaceutical companies stand up a pediatric trial, they get a network going, takes them 2 years sometimes to get it clicking, and then they bring it down because they don't have another product to put in there. It just doesn't seem to make sense. That's one example. And there are a number of other projects that I won't preempt today that we will be making known towards the end of this year. But I'll stop there and turn it back to Mark, and I'll be happy to take some questions later on.
Thanks very much, Delver. I'd like to turn to Jeff next for some opening comments.
Well, first of all, I had the privilege of working with Dr. Hudson during her first term into the federal government at the National Human Genome Research Institute with Dr. Collins and others, and left a decade ago to form TGen, which is a nonprofit biomedical research institute that has brought together physicians and scientists to explore new models and create a culture of adaptive or agile learning to enable molecular medicine to evolve. And collaboratively, and perhaps rather simplistically, we've developed advanced ways of mapping big data to prior knowledge in order to extract the most useful, clinically actionable insights for the patients that sit in front of us today. So the dilemma for saying that, for anyone that says that they're trying to apply today in a clinical context, in clinical trials for patients sitting in front of us today, the use of deep molecular profiling and big data solutions to literally drive medical decisions, is that you get a twofold reaction. One is, there's a group of individuals that say, well, that's no longer differentiating on its own, that even integrating multiple data types and intelligent decision support systems is now ubiquitous. On the flip side, amazingly, there's growing skepticism of the use of precision or individualized medicine, genomically enabled trials, by those that feel that we're already too innovative, that anything beyond well-validated predictive markers is reckless. So the typical reaction ranges from, so what, isn't everyone doing this, to, wait a minute, there's just unproven value, unknown risks. It's too early to have any broad impact. So how do you fit in the framework of that? And I came today really wanting to say that the message is that genomic-enabled medicine is impacting the lives of patients today. It can be incorporated adequately into clinical trials. And the research institute that we've developed, and of course there are many consortiums that have done the same, provide an opportunity of learning to evolve towards transforming medicine and model testing beyond the current models within the healthcare system. So our clinical program at TGen today, which is directed by its physician-in-chief, Dan Von Hoff, recently was chronicled in the Time magazine article that related to the Stand Up to Cancer framework on how to cure cancer. But TGen enrolls hundreds of patients annually in our phase 1 clinical trial program. Those are patients who, as you know, have an average of 12 weeks to live. They're almost all placed on innovative clinical trials to bring together genomics and computational technologies. But importantly, within our single institute, we use that as an iterative learning loop to try to take the information from one patient and bring it forward to another patient. I would echo what Dr. McClellan has published in one of his recent papers on accelerating a pathway for targeted cancer therapies, that in some regards what we're trying to do is move beyond developing evidence-based approaches for subpopulations, something that he wrote about, to again implementing the practice of N-of-1 molecular medicine, where you treat based on the knowledge of a mechanism with evidence coming from the integrated profiling of a single patient. It's not based on epidemiologic information. So I'll just end my opening comment on that with was stating that clearly the mechanistic models we utilize are imperfect. They'll emerge over the time. We don't claim by any means to have solved or created any sort of perfect model, but over the past 10 years, we've involved clinical trial development in this space to create a collective learning and an agile and adaptive approach for delivering science to medicine to individual patients. And it's something that we're excited about being able to share with others.
Great. Thanks very much for the comments, Jeff. Martin?
Thank you, Mark. I feel a little in target here because I think I'm the first person representing a for-profit company that's speaking today. The area in which I work could broadly be called clinical decision support. It's health informatics research at IBM, and we are developing tools to use both structured data, such as from electronic health records, and the unstructured data, the text-like information, such as in journal articles. And the reason we're doing it, and there's been a lot of discussion so far about trials and collecting data and processing data, but the next step is using the data, is how are you going to use what's available out there to make better decisions. And I think we can reasonably say that we have some goals for the future of healthcare, whether you describe it simply as like Don Berwick's triple aim, or talk about personalized healthcare or evidence-supported decision-making efficiencies to eliminate the 1/3 of the healthcare spend in the United States that some conjecture is of no value, is we're going to have to make better decisions. And making better decisions means using the information that's available. The opportunity is that there's a huge amount of information out there. But that's also the challenge. There's a huge amount of information out there, and how can we use it? I've been in healthcare in one fashion or another since the 60s. So starting back in the 60s, and for a couple of decades after that, the challenge was getting the information. So for example, if you had a patient you were managing and you wanted to go and read about this information about this patient, you had a choice, something simple like the textbook, or you would go to the library. And I don't know how many people here remember Index Medicus. You would pull down the volume of Index Medicus off the shelf and think of all the possible categories that might be relevant to your question and go through page after page after page and write down, since there were no laptop computers, write down on a piece of paper the articles you thought were relevant, then take them to the librarian and hope that he or she could get them for you. And you then take home a stack of 150 papers, which actually you could only do after Xerox became cheap enough to be able to do that. Take it back to your home or office, spend a lot of time reading. And the person on rounds in the hospital who was most honored was that person who could remember all that stuff, okay, and shoot it back at you on rounds. Okay. We're in a very different era. Okay. Now the problem is getting all those articles is trivial. Finding which is relevant in them is the real challenge. And I think it's going to reflect what I think will have to be a fundamental change in medical education. Because I started off as an engineer. I went to school as a biomedical systems engineer, electrical engineer. And the emphasis was understanding. Amar Bose, who just died this week, was my first engineering professor. And he said, good engineers know a lot. Great engineers understand a lot and know where to find it. Then I started my first year in medical school, and instead of understanding, it seemed like the educational paradigm was how much rote could I cram into the student's head before they had a seizure. Okay. Totally different. But we are now moving into an era where, obviously having knowledge is going to be of value, but being able to access knowledge and know how to use it, critical thinking, critical analysis, critical use, will be the most revered talent going forward. In that context, developing Watson for healthcare is one of our goals. Probably most people know Watson when it successfully defeated the reigning Jeopardy champions 2 years ago. Fortunately for us in research, the IBM senior leadership recognized playing quiz games was not a long-term business model. We had to move on to something else. So what we do with Watson is look at part of the information you need for decision support. We roughly break decision support into two categories. One we call knowledge-driven. Okay. It assumes the knowledge is out there in print someplace. And the challenge is finding it and understanding what is relevant in that literature and bring it to you in a usable way, ideas that you can use for decision making. The other side we call data-driven decision support, secondary use or reuse of existing data, do population analyses, comparative effective analysis, subpopulation analyses that give you inferences you can use for making decisions about an individual patient or population. So Watson is our knowledge-driven decision support tool. And what it does is it actually understands English, as it showed in Playing Jeopardy. So understand the nature of the question that you have to address, and that question can be expressed in natural language. It can be a specific question, or it can be reading the entirety of a patient's electronic health record. Understanding the question you want to resolve, going out into the literature, and it can be the most current literature, and it doesn't need index medicus, and read all that literature. The Watson that played Jeopardy was able to read and understand 200 million pages of text in 3 seconds, and bring back ideas from that literature in a prioritized way based on Watson's confidence that these ideas are relevant to the decision you have to make, and present those ideas to you in that prioritized way. So it is what is now being referred to as one of the first steps in cognitive computing. Most of the computing that most of us grew up with was programmatic computing. You had a specific set of data that you wanted to mash on or process, and you programmed the computer to process that data. In cognitive computing, the computer you're dealing with big data, ambiguity, inconsistency, statistical inferences, and so you may come up with ideas that you had never even thought about as the outcome of cognitive computing. So Watson does that sort of work. You can ask it a question, it brings ideas back to you. If you want to read more about it, if say Watson comes back with a suggestion that you either don't understand or want to learn more about, you can link on that suggestion, and Watson will take you to the literature that it used to create that suggestion. So in a sense, it's the reverse of a search engine. If you use a search engine, you get a whole bunch of articles and you have to read them and see if there are any ideas in there that are relevant. Watson does the opposite, does the reading for you, brings back the ideas, and then can link you to the articles themselves if you want to do a deeper dive. So it is part now of helping us use the huge amount of text-like information that's out there to help us make better decisions. And it's not a decision-making tool, it's a decision support tool. So it brings ideas to you that it believes are relevant to your decision, and you can use them as you choose. You may decide that some of these suggestions are off the wall and irrelevant, or you may decide that one of these was a great insight that you're going to find very valuable, but it brings ideas to you. So it still requires your critical thinking, okay, which is what domain experts, physicians have, that relationship with the patient, that domain expertise. So Watson is truly decision support and giving you access to all this data out there. For instance, in 2010, the National Library of Medicine cataloged 699,000 new articles. I would love to have said I could read them all, but Watson can and brings the ideas to you. So it gives you access to more information to help you make better decisions. I think we need that now, that with this information explosion, where getting the information is not the challenge, using the information is the challenge.
Thanks very much, Martin. I'd like to turn to Gabriel now.
What does it mean for a patient suffering from MS to have another year walking before they are bound to a wheelchair? What's it mean for a patient suffering from Lou Gehrig's disease to be able to brush their teeth so their caregiver needn't assist them? Sadly, the answer to these questions represent clinical endpoints that are rarely studied in clinical medicine. There's at best a fuzzy answer. Yet, beyond a cure, these are possibly the most important questions a patient suffering from ALS or MS might ask of their clinician. There are thousands of other questions that patients suffering from similar debilitating diseases ask of their doctor, just to be answered with little hard evidence or tremendous uncertainty. The gold standard endpoints of clinical trials rarely quantify the true burden of disease or the hidden benefits of treatment, leaving critical information gaps between the clinician and the practitioner and the patient. At Patients Like Me, we've built a community of over 200,000 patients suffering from life-altering and chronic diseases, including MS, ALS, Parkinson's disease, fibromyalgia, epilepsy, depression, among the other 1,800 diseases we track. Our mission is to help patients understand the answer to a simple yet fundamental question that they are asking to answer. Given my disease diagnosis, what's the best outcome that I can hope to achieve, and how can I get there? Patients in our community voluntarily donate data describing their disease course, symptoms, treatment, and management. We then mine these data for insights to optimize treatment regimens, model disease and its progression, and identify areas of unmet need in the lives of these patients. Not only do we learn from the journeys of these patients, but for the patient, simply participating in our community and learning from the data we've collected ends up improving their disease management as well. For example, in a recent case study we published of 220 epilepsy patients, 59% said they had a better understanding of their seizures, 55% learned about epilepsy symptoms and treatments beyond what they already knew, 28% had better control of their epilepsy, 30% reported better adherence to their medication, and 23% of these people had fewer ER visits, the same ER visits that many people regard as the costliest part of our healthcare system. Ultimately, it turns out there's a whole heck of a lot that we can teach ourselves and teach medicine simply by aggregating the voices of a few thousand patients. Sure, there are limitations to our platform, like we rely on the voluntary donations of data from patients in our communities. Some people discount the fact that it was a patient reporting the data rather than a clinician. Yet our research has shown a very high concordance of the two reporting modalities. Another challenge we face is that most patients who come to the site don't donate a significant enough amount of data to be meaningful. But for the tens of thousands who have donated enough data, there's a lot of good we can do with studying their experiences and journeys. Let me give you one concrete example of this network in action. In early 2008, a small clinical trial in Europe involving 44 patients with ALS ostensibly showed that taking lithium carbonate slowed down the progression of their disease. Naturally, patients in the US and around the world were thrilled to have their first shot at taking a therapy that may help extend their lives and reduce the burden of disease. Instantly, the paper created a tremendous buzz on the chat forums at Patients Like Me, and within 2 months there were 350 patients taking lithium as an off-label therapy they chose to pursue in consultation with their own doctors. At the time, we realized at Patients Like Me that we were in a unique position. We had an opportunity to measure these patients' experiences and reflect back to them the insights that they had collectively generated. So we launched our ALS lithium treatment tracking tool, which enabled patients to join our voluntary study. By 2009, we had gathered enough evidence to be able to publicly announce that there was no benefit of treatment by lithium carbonate to ALS patients. We released the studies within 1 year of the original paper being published and before an NIH-sponsored clinical trial could even begin enrolling its first patient. The cost of our work was a few $1,000s for the software, for the development of the statistics, and for the publication of the results. A mere pittance compared to the multi-million-dollar clinical trial that followed and proved the exact same results. Well, what does the future of medicine hold? Each of the speakers here today have outlined to you some of the ways that they are aiming to improve healthcare and medicine for patients. But for all of us and all of you, I urge you to always remember that why we're in healthcare, and that is for the patient. If we simply listen to the patient's voice and we bring their perspective into medical care of what matters, we can actually do a tremendous amount of good beyond just the endpoints that we measure in our clinical trials. Thank you.
Thanks very much, Gabriel. Well, you all collectively who come from nonprofit companies, for-profit companies, nonprofit companies funded by for-profit companies, have covered a lot of steps that you're taking to address inefficiencies in the research and development processes, overcoming difficulties in obtaining and integrating complex data, getting more meaningful data from patients more quickly. Just as a counterpoint to our first panel, which was about sort of the policy steps and the government funding, government initiatives that are being undertaken to address some of these challenges, I'd like to ask you all, what should the government be doing? I mean, it seems like a lot of the steps that you all are taking are addressing some of these fundamental challenges that have come up today in the efficiency and the comprehensiveness and the speed of the process of generating new evidence. What is the best role for government given all these kinds of evolving and new types of activities that are being undertaken outside of government?
Well, go ahead.
Well, thank you. All I wanted to say was that one thing we've observed in our work with companies in industry and other stakeholders is that companies are very hard to open their ear to patients because essentially they open their ear to all the adverse events that can occur along with listening to such a torrent of information. So despite the fact that there's large evidence to suggest that listening to the patient can add value and innovation, companies are loath to open their ears to that because of all the compliance issues that relate to it. So I'd say at the very least having better understanding and better guidance from the government on what companies need to do with the adverse events or the information they learn from patients and how they can better use that, I think will fundamentally enable them to listen better to the patient.
Okay. Dalver, could you comment on this last point? Because you all are taking some steps that, as I understand it, would encourage companies to get more data from more sources more quickly using kind of standardized ways. And you focused on evidence, but you mentioned that the standards for identifying more and less important safety issues have been worked on that already that you're building on as well.
Yeah, no, I think...
And feel free to comment on the original question, too.
No, sure. Just to that point, I think the landscape is changing, even within industry. I think it is clear that not listening to safety signals, whichever forum or wherever they may come from, is a short-sighted perspective. It eventually will become a problem at some point. So I think that's abating, wherever it is that it may have existed in the past. But in terms of your original question, Mark, I think the government, and I'm not sure which part of the government you're talking about, but I think what we are seeing is great collaboration. And one thing I did not mention in my preamble about Transcelerate is it's not existing as some big pharma club off in some corner doing its own thing. We're actively engaged with multiple parties. We're actively engaged with Citi. I mentioned the Society of Clinical Research sites. We're working with, we've just had discussions with the NCI. I met with ASCO folks yesterday about CancerLink and what they're doing there, et cetera, et cetera. We're working with the regulators around the world. What we are finding, Mark, is the circumstances that I described earlier, where the industry is in a circumstance where the model is no longer sustainable. It's also very well recognized that if that engine slows down, it impacts our ability to get more drugs to the patients that are out there looking for interventions that have still to be, discovered and delivered to patients. So I think you're finding a lot more cooperation, a lot less silo thinking. Francis Collins of NIH came to some of the meetings we've had to discuss areas of collaboration, similarly with NCI, FDA and EMA in Europe, and now with the Japanese and Chinese regulatory agencies there. They're not just involved with us, they're actively seeking to be involved, that the solutions we put out with their understanding, with their guidances, to make sure that we're not replicating efforts. So I think I think we've turned the corner to some extent, where we collectively, doing clinical trials for the sake of patients, realize that the current paradigm is not going to sustain us in the long term. And so you're seeing a lot more collaboration and cooperation from multiple parties than you probably would have seen 5 or 10 years ago.
I think there were a couple of things. One is that the basic premise, at least from the computer scientist colleagues that I work with, is the more data, the better. There's no such thing as too much data. When you have these supercomputers that can chew through anything, there isn't. No such thing as too much data. The more data we have, the more precise our analyses appropriate for an individual patient can be, including patient input, because one of the goals of the future for healthcare is sometimes referred to as the empowered, knowledgeable patient. So a patient who better understands his or her own health situation is a better manager. And the empowered patient is a patient who can actively participate in the decision process, because we're moving away from the paternalistic area of healthcare that I started in, where you do what I tell you because I'm the doctor and I know everything, you know nothing. we want knowledgeable patients. And that's one of our efforts with Watson. We're working with Memorial Sloan Kettering and WellPoint to develop Watson for support in making therapeutic decisions in oncology. And Watson is being taught how to incorporate patient preferences. So we think that that's a key role. But one of the challenges for the future is given, proper due to privacy and security, is getting access to information. Right now, there's so many obstacles to collecting information. In fact, there was a bill submitted to the Virginia Assembly in January prohibiting the use of healthcare information for analytics. I think those social and political obstacles are some of the major challenges that society and government have to help us address.
Jeff?
Well, I think there's two aspects. There is a group of individuals, Kathy Giusti from the Multiple Myeloma Research Foundation, as you mentioned, is a great example of someone who decided not to wait for a relatively rare disease, myeloma, to be put into the framework of one of the larger groups like TCGA, mount their own group effort, mount a clinical trial program, and has really done a remarkable job. We're fortunate to be the group doing the genomic analysis on a 1,000-patient 10-year longitudinal study to capture all that information, and that's really a remarkable testimony to how the patient groups themselves can really mount a tremendous effort. The other project, though, that I'd mention, and the reason why I think the government plays such a critical role is I had a Stand Up to Cancer Melanoma Dream Team with Pat LaRusso that's funded in the area of trying to address randomized clinical trial using complete genomic sequencing and information in support of treatment decisions, in which we actually have been able to bring 21 investigational agents under a single umbrella IND, and to be able to look at that genomic information and bring treatment recommendations to patients across about a dozen different clinical centers. And we've worked closely with Dr. Woodcock in both the CDER and CDRH, the idea that you have to align everything you do if you're going to do these clinical trials, not just with the drug evaluation and research branch, but increasingly the device group. So yesterday we were on the phone with about 30 people from the from the FDA, just finalizing the Plan B design for that clinical trial. Again, not sponsored by the government, but extraordinarily supported by groups like NIST and others in terms of validation cohorts and others. So I think the government coming alongside those is a really important component of where they can add value.
I did hear from all of you sort of this consistent theme of government collaboration, involvement in both identifying problems. For FDA, for example, Dalver, for some years, FDA has been identifying priority issues and their so-called critical path gaps, which seem to fit a lot with some of the areas where Transcelerate is focusing. So helping to bring a focus, helping to bring some attention. It is a somewhat different model than the old just fund the study and step back, or just fund the study and then regulate after the fact, which I think will continue to evolve. I did want to follow up on one, I think particularly interesting to me, set of comments that had come out of the previous session and this one, whereas we're all talking about the explosion of data and the explosion or the potentially increasing opportunities for doing large-scale clinical studies. You heard a lot on the first panel about the importance for confidence in the evidence that was coming out, coming from reproducibility, from using standard approaches, well-defined approaches. And I want to contrast that with some of what I was hearing from both, especially Jeff and Martin, maybe from you all, about the importance of embracing the data explosion and the ends of one, and bringing in data from other multiple sources, which I think as you heard earlier, might raise questions of, can we interpret this? Can we reproduce it? Is it really, say, a causal relationship with a treatment? When you don't have those same kinds of well-defined protocols and standards established, is this something that I'm just seeing as a potential conflict, or is this a real tension in bringing together these different types of data and in supporting consistent infrastructures on the one hand, but bring in wide sources of new kinds of information on the other?
I think you're on target. I mean, in part, if we look at the value we've gotten from, well-controlled studies that are published in reputable journals, it hasn't had the impact one would expect, and it's just as likely that a year later in another reputable journal there'll be another study equally well-controlled that contradicts the first. So, we haven't been in a position where, we know exactly what to do, and so much that I was taught in medical school and residency as being the only way to take care of things has since been proven to be not only useless, but frankly dangerous. So we've always been in that arena right now. That's not going to change. And as I think will happen, a lot more of the inference we draw will be, secondary use or reuse of existing data. And if, big data, it's sort of defined in the mind of the beholder. So it's usually described by its characteristics, which are volume, velocity, because how fast it's coming at you, variety, because of the different kinds, and then the fourth V is either value or veracity. Because when you're dealing with big data, you're dealing with conflict, you're dealing with uncertainty. And so more than eliminating that, because I don't think you will, is the analytic tools have to find somehow of dealing with that conflict and inconsistency. So for example, when Watson processes information, if it finds information, sources of information in conflict, it processes that conflict, that disagreement, and it results in a change in its confidence when it offers a suggestion to you as to how likely this is to be important for your consideration. So since we can't eliminate it, we have to deal with it and let the result of our analyses reflect that ambiguity. As far as the kinds of information, I think Dr. Woodcock referenced one of the limitations of the electronic health records were the constraint in which you could enter information into these structured drop-down menus. And it used to frustrate me using electronic health records that I had to convert all that I knew about this situation into little columns or drop-down. So one of the thoughts I have for the future, and I have to preface it with the remark that many of my ideas and thoughts make my colleagues raise their eyebrows and chuckle, to the point where I have included in my email signature a quote from Albert Einstein for an idea that does not at first seem insane: there is no hope. So over the 30 plus years or so that electronic health records have evolved, it's been more and more aggressively how you can get that information into structured sources, structured memory, drop-down menus or so on. Since we're now doing much better at natural language processing, my hypothesis is at some point that trend is going to reverse. And we will be able to go back to the narrative era of electronic health records because we will have the tools that actually can process and understand that information. We have not before, and that was the driving force for making everything structured because there's the only way we could analyze it. And the narrative part, the scanned reports, were peripheral to what the analysis and the electronic health records could do. So my prediction, with all the caveats I just gave you, is that's going to change and we're going to make better use of that kind of information.
I would like Jeff's comment on this, but just to follow up, since I asked the first panel basically how far are we from having well-structured data and protocols so there could be confidence in these very large-scale research studies. I'll ask you the converse question: how far are we from having those natural language tools that would really give clinicians, regulators, others confidence about being able to bring together much less formally structured data?
We're still very early on. I've seen the toddler stage.
Yeah. Okay.
Well, I agree with the toddler stage. This is a beginning of a new era of implementing this. But again, the design of that Stand Up to Cancer melanoma research trial is just a paradigm as an example. So you've got CDER regulating the drugs that go into the trial. You have CDRH working with us on the validation steps so that we develop a genomic consortium report. We don't use Watson, we use a different approach for knowledge retrieval around the compendium of drugs that would be included. And we set up a series of rules and heuristics that are evolved, and we lock that down at the time of the introduction of the trial so that you're merging the information from your rules database and you're merging the information from your genomic compendium to come up with a structured list of potential options in which for that individual patient they have a change that attaches to a rule that we look at. with Rich Simon and Don Bear, we built an adaptive trial design to be able to actually implement this. So we use the first patient and then the second patient and the third patient. We don't have to repeat the same mistake over and over again. We have an iterative learning loop, and then we have a randomization component in which we'll compare versus standard of care, and then we'll have a crossover option for those patients. So it's building into the design. I actually really liked what Janet Woodcock said, in that you have to design a clinical trial and the framework to accomplish those if you're going to move into that area. And that's really what's driven this type of approach. So very early in this, it doesn't take away any of the importance of a Watson or other component, but integrating those pieces together, people need to begin to accomplish today.
Gabriel, how far do you see patients like me from being able to be the engine for a kind of randomized studies? But I hear what you're saying about being able to get large-scale, pretty structured data from patients about side effects and so forth. But what about randomized trials?
Yeah, it's always difficult because we're not practitioners. We're really observational in orientation for how we approach medicine. I would say that it is largely our vision, though, at least to start with the pharmacovigilance side of things, the phase 4 trials, and to help medicine better understand the implications of large populations and what types of drugs are working for who. But ultimately, we want to work up that value chain towards more types of interventional trials and help patients better understand which treatments are most likely to work for them. So we're getting also more involved with time in the molecular side of it all, and that we recognize that there's a limit to what patient-reported data can provide, and that molecular data and allowing folks to compare their molecular profile of their tumor with that of another patient may actually have a similar effect as them being able to compare their PRO-type data to other patients. So by all means, we very much want to empower the patients to lend not only their experience on therapies or experience with disease, but also to lend their data to other patients as well.
Well, with all this talk about new collaborations and partnerships, maybe in a future event, TGen and patients like me might be working even more closely together. I'd like to open this up to some comments from all of you. So I see some hands up here and at the back.
Hi, I'm Julia Manning from research think tank 2020 Health based in London. I've got a question for Dr. Gill. I just wondered if you are extrapolating the potential savings from the standardization of protocols that you are developing with the different companies that you're working with. And also a question for Dr. Eichler. A lot of your focus is on the behavioral outcomes for patients, and I wondered if you have noticed anyone else picking up on that. So Dr. Luce's patient networks, whether they are actually focusing on behaviors, for instance, being able to walk your dog, rather than keeping your HbA1c at a certain level. So things that are meaningful for patients.
Do you want to go first?
No, you, please.
I think your question, if I make sure I'm answering your question, what are the implications in terms of cost savings? Is that your question? So the short answer is yes. In order to embark on an endeavor like this, the member companies obviously need to understand that it's going to have a meaningful impact, that it's not just exercise in optics. And clearly there are quantitative and qualitative benefits from what we're doing here. The quantitative benefits, the qualitative benefits, certainly from the industry perspective, that are harder for them to quantify are, for example, what is the value of investigators not having to spin their wheels, redoing work and requalifying themselves? So many of those benefits. But from a quantitative perspective, yes, there are enormous amounts of money tied to just the 5 initiatives we've launched so far, in the many, many hundreds of millions of dollars in potential savings, and in only the Transcelerate member companies, which don't make up the entire R&D spend. They make a significant portion of it, but they're not the entire R&D spend. They're over 50%, for example. So yes, and the stated goal of these companies is not to increase their margins with this. To the company, the commitment is to plow these savings, so to speak, back into developing the drugs that I mentioned earlier. Many of them have, but can't develop because they just can't fund them in the current configuration and cost structures.
And to answer your question, Patients Like Me was founded with a perspective on focusing on neuroscience, neurological disorders initially, with some of our earliest community work based on ALS. So that's sort of the history of where this PRO side came from. And we think that especially inside of neurological disorders, patient-reported outcomes have a tremendous opportunity of really enhancing the phenome, if you will, of the patient experience. But I would very much agree that beyond neuroscience and neurological disorders and degenerative neurological disorders, when you get into things like cancer or other elements like diabetes, as you mentioned, there's need to augment the patient-reported elements of the patient experience with additional data. And so that's why, as I said, we're getting better and more interested in collecting additional data that patients themselves can collect and help patients articulate the journey they've been on by showing numbers beyond just those that they've reported, but rather some of those labs, the omics type things. We recognize that if a patient is interested in exploring an off-label medicine that they may have learned about somewhere, there's very little opportunity for them to actually find good evidence about that medicine if it hasn't been through some trials, other than perhaps going to TGen. I've been a big fan of the work done at TGen for years, but there's very limited options for how to explore alternative and complementary therapies, off-label therapies. And our hope is that by augmenting the data we collect, we can actually get better at that and hope ultimately help enrich this conversation about helping patients find the right therapy for them.
And we're still, if I may add one comment, we're still, to the off-label comment you made, we don't know the outcomes of the off-label use. There's no systematic way to find out, well, off-label was used, but what happened? So we spin in the cycle of blindness and we continue. It may be totally ineffective in that off-label use, but we may not find out for a lot longer than we should.
Well, this is the hope by allowing that patient to own their results and report on their outcome, that maybe
Yes.
there can be that critical mass. And that's what we did with lithium, which I think was so powerful, is we were able to put to rest a poor use of an off-label therapy.
Thank you. In the back.
It's Joseph Hutter from CMS, and starting with Gabriel, the quality metrics that you mentioned about walking for one more year, brushing teeth without a caregiver help, is actually of interest to us, at least, because they objectively impact the illness or the quality of life that's experienced by the patient themselves. It's interesting that the study that you did for thousands of dollars in less than a year might be interesting if you could generate a lot of those things as hypothesis-generating studies, which can help you target which of these more complicated and prospective randomized control studies might be more worthwhile. It's almost a screening tool to identify what are the studies you want. When you get into those studies then, two thoughts. One's going back to Transcelerate, is that you want to streamline those studies, and there's a lot of the stuff that you do for streamlining studies, not just logistically in terms of let's have one standard for IRBs, but also in terms of how can we sort of put adaptive trial design along with randomization in a prospective study to get more bang for your buck and squeeze the timelines for development. And then finally, where do you interject some of those outcomes that you're talking to? That goes back to Kathy Hudson, I think. We have something that's CMS FDA parallel review. Let's get CMS interest in outcomes there at the table so that companies can look at those and address them when they're going to the FDA. In some cases, might it be like a triple review where when you're designing some of these studies at NIH, does it make sense to have some of those clinical outcomes and quality metrics in mind when you're designing those studies? That would be really interesting.
Comments? It really is about, I think, integrating the kinds of patient-reported outcomes that you've been focusing on throughout the whole research and development process more efficiently and consistently.
My one comment to you is I couldn't agree more with your perspective and your enthusiasm behind those ideas. Absolutely. In fact, that is the way we work with a lot of our corporate clients. They come to us and say, hey, we want to explore the feasibility of this or understand the patient subpopulation or build a trial around these initial hypotheses. And we'll help shape their hypotheses through our research of the data and through the insights we generate. And that ultimately goes to inform their clinical trial design and what they go after.
Question over here.
Hi, my name is Purnima Oraganti, and I'm a medical student with the American Medical Association. My question is for Dr. Eichler. Given that the research conducted at Patients Like Me is different in many ways from a lot of the other research that is done in industry currently, randomized clinical trials or other observational studies. I was just wondering, what is the perception of these studies in industry? Are people utilizing the research and the outcomes of these studies in the same way that information given or taken from a randomized clinical trial is given? Is it utilized in the same developmental way if people are developing new drugs or guidelines for medical practice? So any comments on that would be appreciated. Thank you.
Yeah, sure. I think the reality is that there is a certain amount of discount factor to the evidence we can provide to the conversation, in that we're not a clinical trial. We're not going to be equivalent to the gold standard clinical endpoints. But at the same hand, I think that we don't often strive to be. We want to be an agitator in this environment that helps us suggest new trials that would benefit these patient populations, or helps to highlight some of the, like I said before, the hidden benefits of some of these treatments. One conversation I've had recently with some of our clients and some of our internal stakeholders is around the fact that the gold standard of a clinical trial in cancer is about overall survival typically, and that endpoint fails to take into account all the benefits of lifestyle, of quality of life that can occur in all the time surviving, but rather just looks at this difference in survival time. Well, it means a lot different if your last 8 months on earth are spent at your child's graduation or at a wedding rather than bedridden. And these are the kind of evidence and data that we can bring and hopefully impact clinical trials. But ultimately, it's going to take well-designed clinical trials with really good endpoints to help articulate the benefits and quantify the benefits of these therapies. And that's what I think we'll leave that up to the clinical trials for now. We're not trying to go there quite yet, but ultimately with more numbers and we talked about these standing clinical trials, these standing registries of patients. With more numbers, you can do a lot more in that regard, and that's something we think we'll get into in the coming years.
One of the strategies in analysis of big data is called non-hypothesis testing, and I think it responds to the question of gentleman from CMS and from you. Is there's huge amounts of data out there that are collected for general purposes, not for any specific study. And can that data be used to get valuable clinical inferences? So one of the methodologies we use in data-driven decision support is called patient similarity. You describe your patient with many, many characteristics, potentially thousands of characteristics, and then look in the population of patients for which you have information. So if you're a large integrated network, it may be 10 or 15 million. If you're a European country, it may be 40 million patients about whom you have information. You look at that data and try to define a cohort of patients that are substantially similar to your index patient, and then do look for differences in outcome. So it gives you the opportunity, for example, apropos of your concern about cancer, is defining different endpoints and analyzing those populations from different endpoints and drawing these inferences. And it might also address the off-label use, because if it's in the electronic health record that a patient was taking the drug, you can draw inferences. Now, one of the challenges to non-hypothesis testing is there's a big gap between statistical association and causality. But at the very least, that might be a way of identifying associations that would be more targeted for a specific study on that particular issue. At the worst, at best, it actually gives you inference that you can use for taking care of the patient. So you can read all the literature, and you have this patient, and you have found in the population in which this patient exists, patients like this patient that get this treatment instead of that treatment do better. It may not be in the literature, but you may actually draw real information. So this non-hypothesis testing is fairly powerful with limitations, but does start to address some of the other issues of concern, and certainly allows you to address more topics than coming up with a whole bunch of specific randomized double-blind controlled studies you set up going a priori.
I think this last discussion, which is combined patient-generated and traditional outcome measures and randomized versus observational studies and unstructured versus more structured data, is a great point to end this particular conversation on. Clearly, you all have brought out a lot of the dynamics and changes that are happening in research and development and evidence development today, and I want to thank you for a really interesting discussion. Thank you. Now, We are going to take a 10-minute break, reconvene at about 11:25, so I'd ask you to be back in your seats by that time. Thank you very much. A lot more to discuss. Our final panel for this year's conference is on establishing metrics and interpreting those metrics for the challenges around biomedical innovation that we've been discussing so far today. And as you have heard, there are a lot of issues, a lot of dynamics in the process. And without ways of measuring what's working and what's not, it's awfully hard to conclude whether we're making progress and what the most promising next steps for progress should be. So that's the focus of this last session. And just as a reminder, I started out this morning with an update on some of the measures that are available now, the most recent measures on what's going on with the productivity of research and development and the magnitude of its impact. The middle of this slide is a reminder about those traditional measures of productivity, things like project approvals, average development time, and average development cost. In light of some of the discussions that we've had today, though there's some interest in further development of ways of knowing whether we're making progress towards some of these traditional goals and whether those traditional measures can be augmented by approaches that could better capture the value, the impact on meaningful patient outcomes that you heard about in the last two panels. So in terms of scientific and development measures, validated biomarkers, failure rates between clinical phases, impact of different kinds of government grant mechanisms are all measurement areas where some research and analysis is being done, as well as more measures directly of the impacts of innovation, impact on population treatment, impact on the outcomes of those patients as well. So we have a framework in mind for thinking about progress and innovation in this more comprehensive way. We'd like to talk about that a bit with this panel. One of the topics that came up in the earlier discussion today in terms of getting at whether real progress is being made, particularly in areas that smaller startup companies may have an impact, was the FDA expedited review processes, including the breakthrough process. These are processes that are used by FDA for treatments that are intended to fill an unmet medical need or that provide a significant advancement in medical care, or that are using newer validated endpoints to demonstrate benefits over current treatments in potentially faster and more efficient ways. The newest of these expedited mechanisms, the breakthrough therapy designation, was established last year and already has been applied in 20 cases by the FDA this year. 17 of those have been made public. Like these other expedited mechanisms, this is a designation granted to investigational products for serious or life-threatening diseases that show considerable early promise in clinical studies. They've been granted in a wide range of areas, about half in oncology, but in a number of other areas as well. So again, maybe another way to help refine our understanding of what is going on with biomedical research and development productivity, and better ways to assess and support the kinds of innovative policy ideas that you've heard about, and the innovative approaches that companies, the private sector, and public-private collaborations are implementing right now. So joining us to provide some more insights around better measurement to support research and development, and to help us interpret the evidence that's out there from the academic community, from the investment community, from other places on R&D productivity and what can be done to address it, is a final distinguished panel here today. We're going to hear from Jonathan Leff, the chairman of Deerfield Institute Institute and partner in Deerfield Management, Murray Aitken, who's the executive director at the IMS Institute for Healthcare Informatics, Ken Caton, professor and director at the Tufts Center for the Study of Drug Development at Tufts. I mentioned some of his studies earlier, and Bernard Munoz, the founder of InnoThink Center for Research and Biomedical Innovation, who himself has compiled a lot of evidence on innovative practices or the lack thereof in pharmaceutical research and development. So we're very pleased to have all of you here with us today. I think I will start with or just go in the order that I just described. So Jonathan, starting with you.
Sure. Thank you, Mark, and thank you for having me here today. I'm Jonathan Leff. As Mark said, I'm a partner with Deerfield Management. I've been an investor primarily in biotechnology companies from a venture capital perspective for more than 17 years now. I also serve on the board of the National Venture Capital Association and chair the life sciences effort of the NVCA, which is called NVCA Medic. So hope to bring some of the perspective here of the investment community, capital formation for supporting innovative R&D in biopharma and medical devices, and perspective from the venture capital side of the world. So metrics for biomedical innovation is the subject, and I guess I thought I'd start just by saying if we want to measure this thing, we at least should start by having a common understanding of what we mean by it. So I'll take a stab at what I mean by innovation, and the other folks here on the panel can confirm that they're talking about the same thing or tell me they're talking about something entirely different. But I think what we mean, or at least what I want to talk about, is new products, new drugs, diagnostics, and medical devices, or potentially new uses for existing products that meaningfully impact health outcomes. And so the measure of whether something is innovative is not simply whether it's new and gee whiz, scientifically exciting, but whether it has a real major change in the impact on the health outcomes of patients. And if we want to measure the effectiveness of this whole scientific and medical R&D enterprise that's built up around translating scientific insight and science into medical products, what we're really talking about ultimately is we'd like to measure how much impact we're having on health outcomes. That's obviously an extraordinarily difficult thing to do, and there's not any single metric or even set of metrics that tells the whole story, which is why Mark showed the slide with a list of things that one can look at and try to piece together a picture of how effectively this enterprise is working and how much impact it is having on health outcomes, and then what one could do to make it work even better. So I've provided two slides that I'm going to walk through today, and it's not because I think these two metrics are the answer or the be-all, end-all. It's really mostly because Mark and the organizers said 2 slides max, and so I had to choose two metrics to put up there, and these are the 2 charts that I chose. And one of the things I'll say about this is we're really all feeling parts of the elephant as blind people here, and every metric that you look at is just one piece of the puzzle that maybe helps provide some context for a picture of how the whole enterprise is doing. So that's the context with which I've chosen these 2 metrics. Also want to say, as we get into these, you'll see both of these pictures that I'm going to show you point pretty much clearly down and to the right. And that's not the direction that we want them to be pointing. So these charts might suggest that I'm pessimistic about the state of the enterprise. That's not really true. I think there are many reasons for optimism and many reasons to suggest that things are better today than they've been in quite a number of years. But these charts still are what they are. It's the data, and we need to grapple with how we got to where we are and what it means for how to make the system work more effectively. So with that, I'll start with the first of these 2, which is not a data set that I created. It's pulled from Scannell et al. Paper in Nature Reviews a little bit more than a year ago, where the authors looked at the productivity of biopharma R&D using a fairly simple measure, which is the number of new drugs approved each year per billions of dollars spent on commercial R&D in that year. And they looked at this simple metric going back 60 years. the result of that is shown here, and it's a pretty striking and remarkable picture. I think many people here have probably seen this before, but it essentially shows a more or less linear decline in the productivity.
And this is a linear decline on a logarithmic scale.
A linear decline on a logarithmic basis, and these are in constant dollars. So the productivity has, you look at this, and I think it was last year at this conference, Francis Collins showed this same chart, and he said, this looks like such a consistent picture, it almost looks like a law of nature. And obviously he didn't mean that. This is not a law of nature, but there are some powerful laws of economics that underlie this, that this picture helps to illustrate. But basically the author showed that the productivity by this measure has been cut in half every 9 years for the last 6 decades, which is this logarithmic decline, which astoundingly means over 60 years you see about an 80 times decrease in the productivity by this measure. And then when you try and decompose what's driving this, it really comes down to 3 things which have been talked about here today, and Mark referenced them in his opening comments and throughout some of the comments. And that is, first and foremost, the time and cost of drug development, that over the decades, it's taken longer and longer and cost more and more to get a single drug approved. And related to that, the probability of success, or the failure rate, has gotten worse and worse over time. Now that changes, depends what data set you look at, but certainly the time and cost escalation has been a pretty steady phenomenon over a period of many decades. So that's the first kind of picture of the system that I wanted to show you. And the second one, which in some ways I think flows from that, is to look at what I do, and that's venture capital investment in biomedical startups. And this data is taken from data published by Price Waterhouse and the National Venture Capital Association. Mark showed some data earlier showing that total venture investing has declined from about $6 billion to $4 billion in life sciences over the past 5 years or so. This is a different slice on the data. This is looking only at first-time venture financing, so startup companies getting their financing for the first time. And I think this is, in many ways, the leading indicator for what's happening in the venture sector, because if new companies don't get started, then there won't be follow-on rounds and ultimately less innovation coming through the funnel from the small company side of the spectrum. So this picture is also pretty daunting. I mean, if you look, focusing, maybe a little hard to see here, but focusing on the green lines on these charts, you see on the left biotech and on the right medical devices, showing more or less the same picture, and that is that the number of new companies getting first-time financing has declined pretty steadily since 2006, 2007 timeframe. And I've annualized the first quarter of 2013. The second quarter data aren't out yet, but the first quarter of this year certainly suggests that we're off to a trajectory that is continuing this down into the right trend. So this is something that is of real concern, of course, not just to the venture capitalists who do this stuff, but it suggests that capital is being directed away from innovation in this sector and to other things. And I can tell you among those other things are investment in technology, social networking, energy, and a whole range of other things, but ultimately means less investment in new medical innovations. So I think the major point that I want to bring out with these 2 slides is that when I look at the innovation enterprise, I see it not so much as, I see it not entirely, but in substantial part as a for-profit enterprise. Now, certainly there are pockets of it that are government-sponsored or philanthropic and so on, but a major share of the dollars that finance drug development in particular is for-profit, and therefore it's shaped by economic forces. So no one in this room or anywhere else should be surprised that when you see this logarithmic decline over 60 years in the productivity of the enterprise, that you start to see pressure on the investment activity that flows out of that. It's simple supply and demand. If you hold everything else in the system constant and make it more expensive to produce something, the something here being new drugs, then you're going to shift the supply curve to the right and you're going to get less of it. And so the remarkable thing really is not so much that we're seeing the pressure on investment. The remarkable thing is that it's survived as long as it has in light of the sort of 60-year decline that we looked at in the prior slide. And there's a whole set of reasons we could talk about another occasion as to why that might be. But I want to close by saying, coming back to this point, that despite all this, I am optimistic. And you might ask why. One reason is that there is some more recent data that suggests that these metrics and this productivity measure is beginning to show some improvement, and maybe we're not going to continue this logarithmic decline. Certainly, if you look at FDA approvals the last couple of years since the data in that Scannell chart was put together, has looked quite good. We've seen more approvals, and we've especially seen more fast-track type approvals of the more important and innovative kinds of drugs. And there's some more recent evidence out there that the failure rate has improved, particularly in the cancer area, over the past number of years. I also believe, and this is hard to prove, but it's sort of in looking at what I look at every day, I believe that the scientific underpinning of what we do in all the decisions that we make about what products to develop and how to study them has gotten stronger. And that's particularly true in certain disease areas like oncology, where we're understanding the molecular mechanisms and the right patient groups to study and so on much better. And that is leading, I think, and should continue to lead to better success rates and shorter development paths. And finally, I would say that the emphasis in the community, in forums like this, and in discussions that have been had over the last number of years within industry and at government in places like FDA, is really taken a much sharper focus on this issue and on understanding that time and cost is impacting the health of the enterprise. And we've seen some real changes. Industry doing things better, doing things smarter, initiatives like Transcelerate Pharma, which has been discussed here today, and also FDA taking real steps that have had significant impact, like breakthrough therapies, like the structured benefit risk framework and the broader application of accelerated approval, all these things designed to get at this question of how do we get drugs that really make an impact on patients through the system faster, less time and cost, and with a better success rate. So I'll stop there.
Great. Thanks very much, Jonathan. Murray?
Thanks. So let me perhaps bring this back to how do we think about establishing baseline metrics that will enable us to say a year from now or 2 years from now or 5 years from now have all the things that we've been talking about this morning really yielded some positive results, and have we moved the needle in terms of the health of the biomedical innovation system. And I bring the perspective from IMS Health, which is a global company that gathers a lot of data, both around biopharmaceuticals, but other parts of aspects of healthcare as well. We do it in over 100 countries, and we've been doing it for almost 60 years. So in many ways, we're the original big data company in healthcare. And I think what we see is still that with respect to biomedical innovation, we haven't made the progress in developing more meaningful metrics than the ones that Mark, with all due respect, showed at the outset, which is how many NCEs are being approved and how much money is going into the R&D system. I think what we recognize though is using metrics is an essential way to know whether in fact we're making progress or not. We also don't need to reinvent the wheel. I think there's a lot of metrics that are out there. They just happen to be in different places around the system, and they never quite come together. And I think the other principle that we always apply is a pragmatic one, which is don't let the perfect be the enemy of the good. We don't need a perfect set of metrics to progress from where we are today in terms of how we look at biomedical innovation. But the metrics we use do need to have some characteristics. They need to be timely. Things move very quickly, and I'm always somewhat dismayed to see references, using data that's 2, 5, or even more years old, when in fact, and again, as we hear this morning, and even looking at the breakthrough designations, how rapidly some of these things are actually moving. And if we don't use timely data, we keep thinking, we're still stuck in the old paradigm. There needs to be some level of objectivity about the data, and so we've always got to be careful about the underlying source of the data and reconciling where there may be conflicts between different sourced data that may relate to objectivity. We have to have the right balance between simplicity and meaningfulness. And again, I think this morning we've heard a good amount of discussion about what things may look like at the totality, at the total level, and how that then looks at a more granular and segmented level. And I think as we understand more about healthcare and more about patient segments and so on, that more granular level is always needed. I mean, I think broadly we look at a set of activity measures that can be very helpful in thinking about innovation. Certainly one of them is the amount of investment that's going in, but also what's in the total pipeline of innovation. There are third-party sources. We have one, but other companies also maintain data sets that are imperfect, though somewhat, that are useful in terms of tracking the totality of what's going on across the industry's R&D pipeline. We have a data set that includes over 3,000 products that are somewhere in clinical development, another 2,500 or so that are in preclinical. We have teams of people who update these data on a daily basis, and that's a useful way to monitor in real time what the current state of the pipeline looks like. It is also a way to identify where there are sort of hot areas of interest and activity and innovation, particularly those focused around specific biomarkers that might be getting a lot of attention, and where we can see these hotspots, if you like, of activity that may indeed be related to efforts to try to stimulate and promote focus in those areas. And then obviously there's the success rates, the transition rates from one stage to the next in clinical development. Again, these are relatively straightforward. The data does exist. It's out there. It's available. It doesn't quite come together. But I think more importantly is going forward are the outcomes measures, because that's really where we can talk about the value that this innovation is bringing patients. So, typically a lot of measures sort of stop with FDA approvals. We would say, well, at least we should look at whether those drugs entered the market. And in the case of 2012, we had a lot of approvals showing up in the last half of December, which didn't actually make their way to patients in 2012, which we think is a more meaningful metric. But even then, we see actually a growing gap between drugs that have been approved and are available through commercial plans, but not yet available through Medicare Part D plans. We've been watching this over the last 2 to 3 years, where we seem to have a slowdown in the time it takes for Medicare Part D beneficiaries to get access to drugs that are approved and are on the market through other plans. Access is a necessary condition, but it's also not sufficient. We also need to be looking at the way in which the drugs are being used. Are they being used appropriately by healthcare professionals? Are they being used appropriately by patients? We know that there's a lot of issues out there, starting with patient non-adherence, but including medication errors and so on, that are a reality of the way in which drugs are used, and which prevents those drugs from actually delivering the full value that is possible. And that then links to the overall sort of outcomes measures from a disease perspective, from a patient segment perspective, where there's a growing amount of data, and again, we heard a lot about the big data explosion. That data has great application in terms of early stage R&D and innovation. It also has great application in terms of us being able to measure the extent to which these new drugs are actually delivering improved outcomes and indeed lowering costs of overall healthcare for the patients who are receiving them. So that measure also, we feel, needs to be brought into the equation as we look at a set of meaningful metrics to measure the state of the innovation system.
Great. Murray, thanks for that comprehensive overview. Ken?
So I run a metrics group, and so this is a good place to be, although I'm a little embarrassed to say that although I run a metrics group that for the last 37 years has tried to get a fix on what represents bioinnovation, we really don't have any idea. So just to highlight some of the things we've already heard about today, just actually for background, my group is a academic-based group. It's the only group that looks at the economic, legal, scientific, and political issues that affect pharmaceutical and biopharmaceutical development. We've been in existence for 37 years, and some of the things that we've done to try to highlight what represents innovation is to look at some of the obvious metrics that you've heard about already, the numbers of approvals, therapeutic outcomes, economic outcomes. We've even looked at patents. We've looked at things like global diffusion of products as an indicator of how valuable that product might be in a global marketplace. And we've looked at other issues as well. And I think the most interesting thing about all of this is that you can draw very different conclusions about whether the state of innovation is positive or negative based on what measure you're using to measure that. So if you look at something like numbers of approvals and you go back over the literature, you get about a 50-50 spread about those that say things look really good and those papers that say things look really bad. But if you look at some of the other metrics like therapeutic outcomes and economic outcomes and even patents, which is probably the messiest of all of them, you get a pretty uniform analysis that basically the state of innovation is on the decline. Things are not as good as they should be. So just based on that, my conclusion has to be that there really is no one measure of innovation. It has to be a number of various factors that we study on a regular basis. We try to do it as timely as possible, as Murray said, and try to get the most accurate data possible to get a fix on what is the current state of innovation or what are the indices that are affecting innovation. The thing that is of greatest interest to me, and I'll just sort of finish with this, is what are the drivers of innovation? What factors are influencing the developers of innovation or the developers of new products to move forward in a particular therapeutic area? And as I look at the landscape and I look at, since we work closely with developers, regulators, and policymakers, I think the four areas, at least in my mind, that are the most important are medical need, of course. Now, that may not necessarily represent a big market, but medical need, the competitive landscape, what other products are out there, how will your product be used, whether there's generic availability, therapeutic alternatives, whether the drug is going to be long-term use or short-term use, and whether you can achieve premium pricing on your product. Scientific knowledge, what's available, what we know, Jonathan sort of alluded to that as well. Along those lines, one of my major concerns is that with all of the emphasis within the NIH now on translational science and translational research, and I can see even within my own institution, but of course within the other academic institutions that I travel to and speak at about partnering with industry, there is such a tremendous focus on translational research, science, or medicine, whatever you want to call it. But the fact of the matter is the fuel that drives that is basic research, and there's a steady starving of the basic research that's going on in a lot of these institutions, which I think eventually is going to force that translational research engine to sort of grind to a halt. And then the last factor, I mentioned the four factors, the last factor I think is just the R&D challenges in general. And if we use as an example of that, we've heard about the CNS diseases. Just to update some of the data that Mark presented at the very outset, our most recent data on the overall cost of drug development based on 2010 data is $1.3 billion to bring a successful compound to market. We are in the process now. We have all the data in. We're preparing our newest paper on what that cost will be. The overall timelines, if you look at just CNS products, is 9.1 years for clinical development and 1.8 years for FDA approval time on average. So it's almost 11 years, just about 11 years to bring those products to market, a tremendous technical risk. And the overall success rate based on 2010 figures for products across the spectrum is 16%. And in particular, if you look at the CNS products, it's sort of a very poor 8% for those products. What's most significant about the CNS products is if you look at that 8% and you look at where those products fail in development, about 60% of them will transition from phase 1 to 2. About 33% will transition from 2 to 3. But then here's the worst case scenario for a product developer. Only 46% of those will transition from phase 3 to approval, which means that 54% of these products are failing in phase 3. And when you look at the timelines that I mentioned earlier, 9.1 years of clinical development for these compounds, what that means is that they're actually failing almost 7 or 8 years into clinical development, which for most companies is untenable situation for a product development plan, which is leading many companies to rethink their strategy in this area. And companies like Sanofi and AstraZeneca and Merck and GlaxoSmithKline have already announced significant reductions in their investment in the CNS space. So what's the conclusion in terms of the state of innovation? Well, I guess like Jonathan, I'm optimistic about it too. And the reason why I'm optimistic about it is I think the industry has hit the lowest point possible in this area, and there's nowhere to go but up. So that's sort of a lame way of saying that it can only get better. But the real conclusion in this is that there is a significant need for change, and I think some of the consortia, some of the partnerships that we're seeing, the Alzheimer's Disease Neuroimaging Initiative, the Mass Neurosciences Consortium, some of the other partnerships that we're seeing between academic groups and industry, as well as industry and patient groups and academic groups, and big pharma consortia, as I mentioned before, venture capital groups and pharma companies collaborating in ways that we haven't seen before. All of that, a new strategic approach, combined with the fact that there is a much greater realization than I've ever seen in my 27 years of working in the group, that the process of clinical development has to be fixed. It's no longer sufficient to just develop drugs the way they developed it in the 1960s, but there really needs to be a way of changing the process of drug development, functional changes, operational changes in development. I think all of that together suggests that somewhere down the line, there's a sunny horizon.
Okay, thanks very much, Ken. Bernard?
Yes, I would concur with a lot of what has been said today, but I take a slightly different perspective on it. I do not dispute that we have lots of metrics, none of which is perfect, to describe innovation. They tend to tell the same story, though, when you put them together. And it is a story of an industry, a great industry, that somehow has lost its touch. If you go back up to the end of the 20th century, pharmaceutical biotechnology was really one of the greatest industries on earth. And rightly so. It has created enormous value for patients, for society, enormous wealth. And today, somehow, this is no longer the case. You've got all the quantitative evidence that is being reflected in the metrics. You also have the qualitative evidence. Great companies that are not so great anymore. Look at Merck, look at AstraZeneca, look at Lilly, look at just about every, Bristol Myers, look at everyone. And it begs the question, why? The ingredients of innovation haven't changed. If anything, they've got better. We've got great scientists, we've got great science, we've got lots of money, and we've got great infrastructure. We've got everything that we need to produce great innovation. And yet, it's not happening, or certainly not happening enough. And it begs the question, why? And how do we react to that situation? And if you look at the way the industry has reacted, it's been somewhat disappointing. It's been haphazard, in my view. So there were fads. For a while it was, well, let's merge. That's going to fix it. And then when it didn't fix it, then the mantra changed. Well, let's demerge. Let's do Six Sigma. Let's do best practices. Let's reorganize. Sometimes let's change the CEOs. And what is troubling about this is that there was no reason in the first place to believe that this or that would work. It was not what I would call an innovation, an evidence-based innovation model. It was basically a gut feel-based innovation model, and the gut feel wasn't very good. So I think we can do better. One of the questions that the industry hasn't been willing to address to this day is, as Ken put it, what produces innovation in the first place? That research has not really been funded. And there has been some of it, though. And if you embrace those conclusions, it will lead you to the conclusion that over the last 15 years, we've had a change of culture in the industry. We've moved from a culture of innovation to a culture of process. And that has pretty much sterilized innovation. Now, some companies have reacted to that. And this is the heartwarming part of it, and because we are seeing progress as a result. You look at Novartis, you look at GSK, you look at Sanofi, Roche, J&J, and the fascinating part is that each one of them has come up with its own solution. It's not like they all listened to the same consultant and implemented the same solution. Each company has found its own pathway back to innovation. Now, some companies are still searching, and hopefully they will find it. But if I look at the future, I think in spite of all the gloom and doom, there is all the reasons to be optimistic. Because, let's face it, this is an industry that is in the process of being disrupted in a major way. And it is disrupted because the current model, whether we like it or not, has led to a massive misallocation of resources. We are spending $135 billion, just industry alone, $135 billion in drug R&D annually. If you add $30 billion in NIH, if you add venture capital, foreign governments, you're over $200 billion annually invested in drug R&D. And for that, what do we get? We get 25 to 35 drugs. Up until recently, those were lousy drugs. Now they're getting better. That's encouraging. But it's still a very poor return for society. We need to do better. And some companies are actually doing much better. If you look at where the value is created in the drug industry today, look at companies such as Regeneron, Celgene, Vertex, Biogen, Onyx. I mean, you've got Genentech, for several decades. You've got companies there that are bucking the trend, that are created tens of billions of dollars in market capitalization when the big pharmaceutical companies are losing market capitalization. Look at Novo. And so if this is typical of industry being disrupted, and we can learn from the research that has studied the phenomenon of disruption, that the industries don't disappear. They morph into something better. Unfortunately, the forces or the companies that do the morphing seldom are the incumbents. And we're seeing that in the pharmaceutical industry today. The companies that are emerging didn't exist a couple of decades ago. And the companies that existed a couple of decades ago, many of them no longer exist. So we see that process of transformation taking place. But one thing is sure, and that is biomedical innovation is not going to disappear because of the entrepreneurs that we've got out there that are hell-bent on making sure that, we're going to have some, because of the society's desire to get innovation and to pay for it. So somehow it's going to happen. It's a bit messy, maybe more than we would like we would like it to be, and there will be casualties. I mean, we've lost 6 big pharmas since 2000, and we may lose another 2 or 3 before it's all said and done. But biomedical innovation will survive.
Thanks. It's interesting that there's a note of optimism throughout the panel, despite all of you talking about metrics that were not uniformly optimistic. I'd like to pick up a little bit on that in the discussion, maybe starting with Bernard's emphasis on company-specific experiences. So a lot of the discussion we've had today have been about broader trends. A lot of the metrics have been sort of industry-wide, but Bernard highlighted that some companies seem to be doing much better than others, and I'm wondering if you all could comment on is there a more systematic way to describe that, or is it are we just stuck with a limited number of case studies because of the size, the limited number of drugs and successes involved? It is a pretty small numbers game, and are there any lessons that you learned that you all have seen from successful models of particular companies? Do you want to start?
Well, yeah, I don't think there's a magic recipe. I mean, look at, what research has shown is that innovation is basically a byproduct of 2 things. I mean, it's not that complicated, only 2 things. One of it is bold thinking, and the other one is cross-pollination. So any model that somehow fosters bold thinking and cross-pollination will see innovation as a byproduct of it. So we know that. And if you look at let's look at the big pharmas, the companies that are pulling themselves out of doldrums. You have a company such as Novartis, which started 10 years ago, and basically their mantra is, let's reconnect with our roots. There's only one kind of research that deserves to be done, and this is breakthrough research. And that's what we're going to do, and the rest of it we're not going to do. It's a model that has worked well for them. You take Sanofi. Companies that were in dire trouble back 5 years ago, and they took a completely different approach. They decided to reposition their companies onto areas that were less IP sensitive. So it's diabetes, it's animal health, it's consumer health, it is vaccines, it is rare diseases. It seems to be working for them. You look at GSK, different model again. Let's try to break the big monolith into smaller units that can operate in some kind of like a biotech network of entities. Seems to be working for them. Roche, no, it's genomic medicine, an integration of diagnostic with advances in genetics and the research model that is basically trying to harness all that. So you've got, you've got, and J&J is different. It's, trying to create a network of partners and let them do their own thing and just manage the whole network so that it does produce the innovation that the company needs to move forward. So there's no single recipe. You've got all kinds of recipes, and as AstraZeneca, Lilly, Merck, Pfizer are trying to pull themselves out of doldrums, they too come with their own personalized solution model. So I think there's plenty of opportunities. What we really need is the leadership. Companies that have leadership are doing much better than the companies that don't. If you look historically at companies that have done well, the one that everybody remembers fondly, they were led by not only great CEOs, but also great CEOs that doubled as chief, innovation officers. Look at Paul Jensen at Jensen. Look at Roy Vagelos at Merck. Look at, Art Levinson at Genentech. There are people who are passionate about science, breakthrough science, and made it known and made it very safe for the people working in their group to really push themselves and be very bold, which is what we know produces innovation. I think we need to kind of reconnect with that culture. We've strayed from it, probably out of ignorance. Some of the ideas that the industry pursued in the last 15 years looked like logical and reasonable ideas at first, like this, idea of industrializing the whole research. Well, they turn out to be disasters. Let's not stick with what doesn't work. Let's reconnect with what does work, because we know what does work. We've done it for almost a century.
Other thoughts on metrics and what can relate to these breakthroughs? Yeah.
Well, I think we've also seen in the last 15 years different companies respond to some external forces. One is payers being a little more demanding in terms of the evidence that the drugs that are being brought to market actually bring value, especially outside of the United States, but to some extent within the US as well. And then regulators have also, I think, changed their approach to, in terms of what is required for companies to submit in terms of dossiers to receive approval. And I think those forces have driven pharmaceutical companies to a greater and or lesser extent to refocus their R&D efforts towards more narrowly scoped therapeutic innovations. So this gets tied up with the movement away from looking for a drug that's going to be a $10 billion product in the market when it's launched, to really redirecting the effort and the resources towards finding drugs that will have a distinctive clinical superiority to not a placebo, but the active comparators that are already available to patients, and thereby focusing more on innovations that will end up with narrower labels, more focus on orphan drugs, and we see that in the mix of drugs that are being approved. We've got a lot more drugs coming onto the market with relatively small target patient populations, and I think some companies have responded more quickly to those forces and ended up now pursuing that kind of innovation than others. So I think that's one of the factors that we've seen play out over the last 10 or at most 15 years.
Yeah, I'd say what characterizes a lot of the companies that Bernard had discussed was really the ability of those companies to focus, to really concentrate their efforts in a few therapeutic areas or product classes and eschew those areas where they just felt it was not possible for them to try to make a meaningful contribution. You look at some companies like Bristol Myers Squibb, which was sort of a standard multi-area pharmaceutical company that tried to be, as many of the legacy pharma companies were, covering a lot of different therapeutic areas, really narrowed their focus, and I think has become very successful in a narrower selection of therapeutic areas. And those that are successful are those like the Genentechs, which have really been focused as well, have been able to concentrate all their efforts in those therapeutic areas, take advantage of the science that's out there and leverage whatever other resources are accessible, but to not disperse those resources over a lot of different therapeutic areas.
Jonathan?
So yeah, just couldn't help thinking in listening to your question, which was really what can we try and identify the recipe for some companies that have had real great success in innovating versus other companies that haven't. And I couldn't help thinking, if I knew that recipe, I would be a really, really, really good investor. Because, of course, it's easy in retrospect to point to the companies that had successful drugs that are making a real impact and are really innovative in creating value, and try to ignore the longer list of companies that have been unsuccessful in trying to do similar sorts of things. So I think the way I think about the question is you've got trying to separate, first of all, the public policy side of all this from the internal decision making that individual corporations make in the for-profit sector. And when you focus on the latter there, the question I would ask is, what are the set of incentives and structures in place that are guiding the sorts of decisions that people make? And one, I think, can have faith that the market forces are pretty powerful. And if the market forces are driving people to make the best possible decisions that translate into innovative products, then there are a lot of good reasons why people are going to do the smartest things they can to achieve that. And I think that is a big reason, I think, why small companies have been successful in bringing a lot of the innovation to the marketplace. As Janet said earlier in her comments this morning, FDA is seeing more and more first-time sponsors, and that actually understates the impact of the small emerging companies, because even many of the drugs brought forward by traditional sponsors like big pharma companies may have been acquired or licensed from small companies along the way. So a lot of the innovation really is coming from small companies. One of the reasons is if you're a small company making decisions about investment in a portfolio, you're facing a much less forgiving environment than if you're a big company with a lot of cash flow and you can afford to make some mistakes. And so the institutional, not that big companies don't want to make really tight decisions as well, but the forces that prevent you from making bad decisions are maybe less powerful in that environment. So I would focus on that a bit. I think that same framework also guides some of my optimism because the markets in general are much less forgiving for capital, and the environment that big pharma companies face today is much less forgiving than it was 10 years or so ago, when pharma companies were able to make enormous amounts of cash flow by bringing on the next generation, extending the patent life of a multi-billion-dollar product. That's what they were going to do. But the environment out there is not rewarding that in the same way anymore, and the shareholders in the big pharma companies are not accepting the level of R&D productivity that they've seen. And so I think pharma is facing a set of institutional pressures, which is already driving a greater focus, I think, on the kinds of decisions that lead to innovative products. And that is dovetailing with the other side of this, which is a public policy environment that is also, and a regulatory environment that is also guiding the ecosystem to make decisions that are leading to more innovation. I think we'll have to see the evidence when we look back 10 years from now, but
we'll see. We'll keep putting together the metrics. I'd like to open this up to some questions and comments from the audience back here. If you wait for the microphone, if you don't mind.
In the previous panel, the man from Transcelerate talked about commonality of getting the 17 partners to do common things in the clinical trials. How, and it seemed very positive, but then you referred to you don't want to industrialize R&D. Those statements seem in opposite direction. Aren't the previous panels all about, in fact, industrializing R&D, getting more precision, knowing what we're doing better? And then you're saying one of the failures of the drug companies the last 10 years has been they tried to industrialize R&D.
Yeah, I think this is because, I mean, it's a confusion that needs to be clarified. You've got two parts in R&D. You've got the discovery part, which is what I would call a mad scientist-driven business. And then you've got the development part, which is driven by execution and economics. And so it's two completely different cultures. And one of the problems that industry has had is that, at times it was the mad scientist culture that dominated, and at other times it was the discipline culture that dominated. And you got to balance the two. And very few organizations have managed to do that successfully. So if it's the mad scientist culture that dominates, pretty soon the company gets into trouble because, well, R&D is a mess because the execution part of it is no longer there. And if you regiment everything, then you're going to sterilize your discovery and you're not going to get innovation. And you don't need to be a scientist to run a big pharma, but it helps. And when scientists were basically dominating the leadership of the industry, they understood the need for balancing, those two imperatives, and some did it better than others. But I think the success of the industry through most of the 20th century goes back to scientists who somehow knew what it takes to do science successfully. And in the 90s, the leadership of the industry the scientists lost the leadership of the industry, went to non-scientists who, many of them, and didn't understand much about science. Some still had good instincts. I mean, one of the best CEO in the industry today is an accountant, but he's got very good instincts. And at the same time, you've got PhD CEOs who are clueless. So you've got to put things in perspective, but somehow you need to balance those two cultures if you're going to have successful innovation.
There was a lot of just a push on this, though. I mean, we take the point about the standardization in the development process of the infrastructure for clinical trials. It's more reliable in the data collection and methods that are designed to bring down the cost of trials and validating markers and so forth for patient response that can improve predictability. But some of the discussion we had this morning was about more systematic approaches to the earlier stage research process. In fact, one defense of the NIH investments in translational research is they were seeing a lot of basic research that didn't seem to be either consistently or reliably translating into products. So I do think there's to push a little bit further on the same question, maybe the 20th century models or mad scientist models, the art and creativity of research is changing a bit with these more targeted therapies and emphasis, like you heard in the second panel, on identifying highest priority druggable targets and things like that.
Well, it may, but the reality is that biology is not a predictive science. You know, we'd love it for it to be predictive, and we think it is predictive. And a lot of the processes that exist today in a pharmaceutical industry assume implicitly that it is predictive, but it is not. And therefore, we fall on our face repeatedly. The serendipity still plays a great role in drug R&D. There is 1/3 of the drugs that are coming to market. We still don't know how they work. So we need to be aware that we've got still huge knowledge gaps in our understanding of biology, which is why we don't have predictability. And one of the best things that NIH could do was actually to fund efforts to eliminate those knowledge gaps, whether they are, genomic or, figure out the junk DNA, figure out the epigenetics, figure out how to put the whole thing together so that you can create an in silico model of biology that is somehow predictive. I mean, we had system biology and 10 years ago, and it hasn't delivered because it could not. We didn't really have the information for it to deliver. So we need to get better at that. And I think as we are, then a lot of the things that we dream of will become possible.
Any other quick thoughts from the panel on this topic?
Well, my quick comment would, we need Watson to be mad from time to time. maybe that's the connection.
I guess they're maybe working on that. I don't know. Let's see, time for one more question back here.
Hello, I'm an international development consultant. I don't have medical biology or background, but thank you very much for a very excellent panel. I'm trying to understand just very common sense economics here. When especially two graphs you have shown the productivity decline last 6 decades, and also investment trends. In international development, in my field, we always have to see international comparators, benchmarks. So none of the presentation today is showing that whether Germany or more appropriate US comparator countries, how they are doing with biomedical products or innovations or R&D, or even Japan. So if the trends you have shown, the productivity decline, every 9 years, almost halved during the 6th decade. Maybe this is just a general global supply chain development for any other commodities, not just the biomedical product or innovation. As I understood, now US imports about 80 or 90% of pharmaceutical drugs imported, and also clinical trials, mostly 70-80% are done overseas. But I haven't seen what are the...
So any comments about the globalization of both production and development of at least some parts of evidence?
Well, I mean, I guess I would say that I believe that the picture shown by that that slide, the Scannell paper, while there are many limitations to what it says and how the ability to interpret that data and draw conclusions from it, I do believe it captures a global phenomenon. And FDA approvals is being used as the metric because that's a easy, consistent data set to get a hold of, and FDA is generally thought of as the global gold standard in regulation. But companies that make investments in biopharma R&D and in innovative products are doing so with the global market in mind. And the FDA is here in that data set is a proxy for what's happening globally. And by the way, non-US companies that are making those investments and then get FDA approval for their products to market them in the US are captured in that data set as well. So I think it's a picture that is generalizable to the global environment. There's no question that pharma companies are recognizing the global supply chain, the global aspect of what they're doing and sourcing products, doing clinical trials overseas, and all of that is a factor and potentially a factor in helping to increase the efficiency of the enterprise over time. But I don't think that one can say this is just another industry where the U.S. is losing out and the industry is moving to Asia or to some other part of the world. I don't think we're seeing that. I think to the extent that investment in venture capital or elsewhere is not being made in innovation in the statistics we're looking at here, I don't think it's being made anywhere else in the world either. I think it's going to other kinds of industries and enterprises.
Any other comments on related to globalization and research and development in biomedicine?
Well, I would fully agree. I mean, I think the biomedical innovation is a global ecosystem. So, you know, the actual products that end up in clinical development in the United States are often discovered somewhere else. They're patented somewhere else, but eventually they make their way into a US-based system in order to get regulatory approval and access the market here. There's certainly a lot more activity going on in many corners of the world with respect to biomedical innovation, but I think it is all reflected broadly in the observations that we've shared here.
Yeah, I guess I'm not as sanguine that innovation stays in this country or even in the Western world. You commented at the end of all of our presentations that we all sort of presented dire statistics, and yet we were all optimistic. But no, I don't think anybody really said where that innovation is going to happen. I don't think that innovation is going to happen here. But I think the rest of the world is gearing up to become a research base for a lot of this activity, where it can be done in many respects the way innovation was done in the United States, sort of that mad scientist type of approach where we'll just put a lot of money into this and see what comes out of it. And if something positive comes out, it becomes a global product. Industry, wherever they are, will see that and say, we don't have to be based in these countries to do this. We could be here, partner with some country and some company in that area and get the benefits. And already some of the pharma companies in the United States are engaging in those types of partnerships. So I think the future of innovation may be positive, but it doesn't mean that it's going to be in the Western world.
Certainly is changing. We are out of time. I want to thank our panel for a really stimulating discussion about metrics and outlook on life and optimism. Thank you.
Thank you.
We have had a long day today, covered a lot of ground. I want to thank again all of our panelists for their contributions. This is clearly an area where there is a lot going on and a lot to come, so stay tuned for further work that we're doing in our innovation initiative and further work from all of these groups. I want to give a special thanks to Greg Daniel, Beth Rafferty, John Berard, Joanna Klatzman, Amy Badel, and Larry Kokot, and especially Morgan Romine for putting together today's events. As I said, all the materials will be available on our website, and we look forward to seeing you at future events and further progress on this very important area of healthcare policy. Thank you very much.